National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Good morning, ladies and gentlemen. Thank you to those who have joined the Zoom room and also official YouTube BRIN Indonesia for the second day of ISIBio 2022. I'm Aulia, will be your master of ceremony for ISIBio. Ladies and gentlemen, before we begin the conference today, allow me to read the guidelines of the conference. Presenters and participants are expected to join at least 30 minutes before the conference started. All participants, both presenters and non-presenters, are expected to join as much as possible all sessions in this conference. Please use the virtual background provided by the official committee. Also, please rename your Zoom username according to the following formats. For presenters, you can write down code of abstract or paper _name_institutions for the example P4.3_Senli Octaviana _BRIN. Or for non-presenters, you can write down P_name_institutions as the example P_Senli Octaviana _BRIN. Please mute the microphone, especially during the presentations of the keynote speakers. And in the parallel sessions, presenters should activate the camera during their presentations. Questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom meeting. And when Q&A or chat feature is selected, please deliver your questions according to this following format: name, institutions, the questions or the name of speakers, and also your questions. Every session will be led by a moderator, and moderator will decide which questions is to be discussed or answered. All participants, presenters, and non-presenters should fill the list of attendees via the link given in each session. Attendees are allowed to know or are not allowed to write down opinion or informations that is offensive, racist, or immortal in nature. An organizing committee have the full right to block or remove any attendee who breaks the above rules. Ladies and gentlemen, we will begin the event. Honorable Dr. Iman Hidayat, Chairman of Research Organizations for Life Sciences and Environment, National Research and Innovation Agency, the Republic of Indonesia. Honorable Head of Research Center for Applied Microbiology BRIN, Head of Research Center for Genetic Engineering BRIN, Head of Research Center under Research Organizations for Life Sciences and Environment BRIN, distinguished speakers and moderators, representatives from ministries, institutions, and universities, and all distinguished guests, ladies and gentlemen. Very good morning from Jakarta. Such a great time for me to welcome back every one of you to the second day of the 9th International Symposium of Innovative Bioproductions Indonesia on Biotechnology and Bioengineering 2022 ISIBio. Ladies and gentlemen, ISIBio has been held annually since 2012, and this year is proudly organized by Research Organizations for Life Sciences and Environment BRIN, bringing the theme of strengthening for bioeconomy through applied biotechnology, bioengineering, and biodiversity. This conference is collaborations with Indonesian Biotechnology Consortium and also ASEAN Federations of Biotechnology Indonesia, and supported by PT Berkaniaga Medica and Nutrilab. As the initial of the agenda for today is keynote speech, therefore may I invite Dr. Puspita Lisdianti as our moderator to lead this morning's sessions. But before that, allow me to read Dr. Puspita Lisdianti's profile. Dr. Puspita Lisdianti is a senior researcher at Biosystematic and Evolution BRIN that has interests on biofilm for health and environment, marine biomicrobiology, cacao fermentations, and also functional foods. She obtained her bachelor degree of molecular biology and agricultural chemistry at Tokyo University of Agriculture, Japan, and continued her master and also PhD at the same university, both majoring biosystematics and agricultural chemistry. Throughout her career, she is also awarded Habibie Prize at 2020, Satya Lencana Karya Satya 30 in 2013, and also Satya Lencana Pembangunan in microbiology. Ladies and gentlemen, please welcome Dr. Puspita Lisdianti. Ibu, time is yours to lead the plenary sessions.
Thank you very much, MC Mbak Aulia. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Honorable Chairman of Research Organization of Life Sciences and Environments, Director of Research Center for Applied Microbiology, Genetic Engineering, Biosystematics and Evaluation BRINs, Chairman of this symposium, and all of the committee. Also, honorable speakers and all participants in this symposium, ladies and gentlemen, I am Puspita Lisdianti, the moderator of this morning symposium. Our keynote speaker today, the first is Dr. Muhammad Sahlan. He is as a representative of Professor Heri Hermansyah, the President of ASEAN Federation of Biotechnology Indonesian Chapter and Dean of Faculty of Teknik Indonesia University. Let me to read Dr. Sahlan CV briefly. Dr. Muhammad Sahlan is a researcher at the Bioprocess Technology Department of Chemical Engineering Universitas Indonesia, obtained a doctor and master degree at the University of Agriculture and Technology, Japan in the field of protein engineering, obtained a bachelor degree at the ITB in the field of chemistry, awarded first winner of invention in beekeeping field, ASEAN Apiculture in 2018, top 107 Indonesian Innovator Business Innovation Center in 2017, gold medal IGEM, In 2014, Roseno Award, Roseno Octroi Bureau, and the National Spice Board in 2013, and Superior Researcher Indo Food Research Nugraha in 2011. Research interest is waste treatment, waste audit, resource recovery, biofilm, phytoremediation, and PICO remediation. His Scopus H index is 14. Before the presentation starts, let me inform you how the presentation will go. First, Dr. Sahlan will be invited to present the material for 30 minutes. After that, there will be a question and answer session for 10 minutes, followed by conclusion. Now, allow me to welcome Dr. Sahlan with the topic Development of Research Collaboration between BRIN and Universities on the topic of biotechnology, bioprocess to improve the bioeconomy sector. Dr. Sahlan, time is yours.
Thank you very much, Dr. Puspita, and also the Master of Ceremony, Ms. Audia. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. I think I have to share my screen for the presentations. Let me to share. Did you see our screen?
Yes, sir. It's already full screen. Thank you very much.
Thank you very much. Okay, recently, now I'm a representative of Professor Heri Hermansyah as the Dean because he have another schedule in this time, and he implied maybe today in this time. So I'm sorry he could not present his presentation. Okay, as Dr. Puspita introduced that in this time, I would like to present about the development of research collaborations and between BRIN and universities on the topic of biotechnology, bioprocess to improve the bioeconomy. In this time, I will focus the presentation or the universities in about the Faculty of Engineering, Universitas Indonesia in collaboration with the BRIN. Recently, I'm also head of the Entrepreneur Innovations Unit and also Reverse Engineering Center that have the correlation between this presentation. Okay, talk about our Faculty of Engineering. We already established on 17 July 1964, and in that time we established 3 departments: civil engineering, mechanical engineering, and electrical engineering. Continue 1 year later, we also established 2 departments: metallurgy and material engineering and architecture. In 1985, we also established the chemical engineering department, started by petrochemical department in that time. And in the 1999, we also established the industrial engineering. Recently, we have a 13 undergraduate program, 2 professional program, 12 master program, and 7 doctoral program. So in detail about the study program in Faculty of Engineering Universitas Indonesia, we have undergraduate program about a certain regular program and 11 parallel program and 7 international program. In the professional program, we have 2 professional program. This is for engineer and also architect. And for the master program, we have 12 master program and 7 doctoral program. So this is the detail of the 13 undergraduate study program from the civil engineering and also mechanical engineering. This is the first in study program in Faculty of Engineering. So all the 7 departments, we have the undergraduate and also international undergraduate program. The international undergraduate program, we have the single and double degree. And every department, we have at least another 1 study program. For example, for the chemical engineering, we have also bioprocess engineering study program. That this study program have very related with this topic about the collaboration between BRIN and university in the field of the bioprocess and biotechnology. And also related to the bio, we also have the biomedical engineering. This is for the postgraduate or postgraduate study program. We have 21. For the professional program, we have engineer and architect. For the master program, we have the civil, mechanical, electrical, metallurgy, and also environmental engineering. We also have the master program for this field. And for doctoral program, we have a 7 doctoral program. This is related to the department in Faculty of Engineering. So since this year, our dean, Professor Alihan Mansa, is the new dean of the Faculty of Engineering, and he know that the condition in the world is very accelerate and the change, and also we have make the strategy how to challenge the accelerated change. So this is the condition in our world. The first is about the volatility of the world, uncertainty, complexity, and also ambiguity of the condition in the world. So based on this condition, we need to collaborate and partnership with other institutions, with other stakeholders. We know that we have the now in disruptive era and also new normal. We have global competition and national competitiveness. We have also industry 4.0 and digitalization, and we need the multidiscipline how to challenge our world in recent condition. So we initiate this challenge by the name of the entrepreneur initiative. We have to challenge. We have the good input in our Faculty of Engineering. We have the students, we have facilities, and others input in our Faculty of Engineering. So we have to make the good impact. We have to increase our service excellence, customer satisfaction culture, reputations, glory on the nations, and also revenue for the economic and advancement. So based on these conditions, we decide 3 strategies. The first is empowering engineering entrepreneurship. The second is impactful research and innovations. The third is modernization of engineering education. Based on the 3 strategies, we have 11 excellent priority programs. The first is the organization of multidiscipline engineering, reverse engineering center, engineering seed fund and grant, engineering revenue recognitions, database engineering networks, organization strategic engineering educations. So because we focus on also in educations, we have to increase the quality of the organizations, and also virtual engineering education facility. As you know that the new era need this conditions. And engineering professional program partnership and collaboration package, international academic recognition and engineering student entrepreneur program. So by this 11 excellent priority program, in this time, we focus on the impactful research and innovations by using reverse engineering strategy. So for the impactful research and innovations, we have a platform for the multidiscipline technology development and product co-creation and reverse engineering. This is the new center in Faculty of Engineering Universitas Indonesia. We have to collaborate with the alumni, society, community, and media, government, business, and also industry. So we have the researcher, we have the facilities and also students, we have inventions, fundamental research funding, and also for the local and global market, we have to innovative solutions, and again, revenue income to increase our quality of the output, and asset equity and also license of the intellectual properties. Based on this strategy, we have also the product after we use this strategy in reverse engineering. We learn from this pandemic COVID-19. We have one good example in this case about the ventilator. So by reverse engineering, we have an activity to increase the value of the technology product, either by modifying or by adding features of the technology. For example, in the first 2000s, in the first time, we make the laboratory prototyping one in this stage, and then after about 6 months, we succeed to use to make the prototype 2, and after 1 year, we succeed to production in industry. So this is one of the good example from the laboratorium or from the lab to the industry. We study the original product and we analysis the product and engineer. We make the prototype and we establish the new product by this scheme of the reverse engineering. By this reverse engineering, we make this center. In this center, we collect the demand from the industry and also from the community. We design the product and we make the seed fund also. We give the fund for the researcher and also for the team from the researcher and also from the students that want to make the reverse reverse engineering product, and the output is the intellectual properties. Yeah, we synergy with our another institution is LemTech with the alumni and partnership industry and sponsor. And with another scheme and the license, we also make the startup and incubate for the technology implementations. And we hope we can increase the revenue of the market revenue for the Faculty of Engineering. This is another product that we have succeed in the pandemic era. Especially, this is the product for in health, because in these conditions, the product of health is very useful or very good in the market. The first is the COPN20. COPN20 ventilator is the first succeed the product, and this product is collaborate not only Faculty of Engineering, but another institution and also the government, how to develop the product and accelerate this product from the laboratory to the industry or to the market. Another product is air purifier with non-thermal called plasma method. This is also the reverse engineering product from Professor Bismo that he expert in the plasma technology and apply this plasma to the air purifier. And this product is also already licensed to the company in Indonesia. Another product is gas supportable incubator. It's it's a little bit different with another product. These implementations in the society is the scheme is by the community engagement. This product is the incubator to warm premature babies or low weight birth babies in order to prevent hypothermia. And this product is portable design and very simple for mobilization. That very good with the Indonesian situation. And this is another product for the implant, and another product is like propolis, and in traditional traditional medicine. This is also from the chemical engineering. So I would like to talk about the example of collaboration between Faculty of Engineering and BRIN. This case is about the development of dengue paper diagnostic. As you know that the dengue is common in tropical countries, including in Indonesia. As we know that about 390 million of the people is already infected by the dengue, and about 75% of the citizens in the tropical countries have infected by this disease. So in the 2019, it's about 120,000 people infected. It is decreased in 2020, but I think it's because of the COVID-19, so it's very difficult to diagnostic and go to the hospital. So maybe this is the undetected of the disease of this dengue. So it's maybe the cause by why the number is decreased. So if they treated, the mortality rate is about 2 until 5%. However, if the untreated, it can reach 50%. Yeah, so it's very important to detect in the first stage of the disease. So about 75% of cases are asymptomatic or mild symptoms. Yeah, and symptoms are similar to other febrile illness. And many of them is small diagnosis, and there are 5 serotypes of dengue virus, and the available diagnostic test in our country is a real-time PCR. Is difficult to operate in the remote area. So our purpose solutions, we have to make the simple assay is a lateral flow. I think is already increased also in Indonesia for the diagnostic test. To develop this product, we need 2 proteins. The first is an S1 proteins as a marker or as the target, and as the detector, we need the antibody and also nanobodies. The NS1 is non-structural protein produced by dengue virus when it is entering a human body by this skin, that after the viral particle enter the cell, the NS1 is produced. So in that time, sometimes the body also can secrete the antibody for the NS1. So the NS1 can be used as the target protein for the diagnostic, and the high level of NS1 protein were found from the day 1 and 3, making it easier for early detection. For the first, we collaborate with the Dr. Widoretno from BRIN. This is for her dissertation research project, and we study how to make the diagnostic or protocol by using surface plasmon resonance. This is the scheme of the surface plasmon resonance. We can coating or immobilize the antibody, and the NS1 is here, and we can detect if there is the different, but the diffraction, it will be make the signal here. So this is our result that this SPR by immobilize the antibody specific for NS1, we can detect the existing of the NS1, and this is concentration depend. And we compare between SPR with the other method. This is the golden method, the RT-PCR, and using ELISA and literal and SPR. And we found that with the golden PCR is also same result. And we found that the baseline between the negative and positive result. So because the antibody is also difficult to produce in Indonesia, so we introduced the nanobodies. What is the nanobodies? Are antibodies with the single variable domain located on the happy chain, also shown as PHH antibodies or single domain antibodies. And because this is single antibodies, without a happy chains, so it's very small, and it's found in camelids and the sharks, and the size also about 10 times smaller compared with the antibody. And so the advantage of the nanobodies is stable in the range of the temperature and exit pH, and can be expressed in E. coli. So for the first, we developed the recombinant of the nanobodies by transform the sequence of the protein to the E. coli, by using specific plasmid, and we succeed to transform to the E. coli, and also is expressed in the E. coli, and we found we detect by SDS phage and Western blotting that the protein expression is succeed. And also we using the Western blotting, we found that the nanobodies is succeed to produce in the E. coli. And the last, we start to make the rapid detections by using the lateral flow, by using nanobodies as the test line, and this is for the control line, anti-mouse antibody here. And this is the only result that we found that the nanobodies also can use as the antibody for in the dot plot anti-NS1 for the rapid test in the future. And last, I would like for the conclusion that our Faculty of Engineering have to collaborate with the BRIN to achieve the impactful of research, and the collaboration between Faculty of Engineering Universitas Indonesia and BRIN could be also in academic and research sectors. Thank you very much for your attention. We hope we can collaborate, and we can contact our public relation and protocol office in Faculty Administration Center in Depok campus Universitas Indonesia. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Sahlan, for very informative presentation. Ladies and gentlemen, now are the time for the Q&A session. Please using chat box or raise your hands to give the questions to Dr. Sahlan. Any questions? Okay, if not, I have one question, Pak Sahlan. Bioengineering is must and have to be mastered by biotechnologists, also biologists, microbiologists should master the bioengineering. Because as a biodiversity country, a mega biodiversity country, we have to make a bioproduct by our own self. No other countries would like to teach us, to make a bioproduct. And then, that's a point. And what is the subject? Would you mind to tell us what is the subject in engineering, faculty of engineering and Universitas Indonesia that correlated to the bioengineering?
Okay, thank you very much, Dr. Puspita, for the questions. In the bio-related subject in our the bioprocess and environmental study program and also biomedical study program, we learn about the basics science and also the engineering science. So we learn from the biochemistry, for example, and also we have to learn for the students, learn about the principle of the basic mathematics, physics, and so on. Not only the biology, but the biology is also important, but another is also important related to the engineering basic science. And the second or the third year, we also have the student have to learn about the basic engineering, like mass transfer and separations and so on, and also related to the biotechnology and the bioprocess, like for the cell culture, fermentation, and so on. And in the last year, they also have to take some elections. For example, the election for the protein engineering and for food industry and pharmacy, and also another advanced subject. And in the engineering, we have the two streams. The first is the process streams. So how to process the bioproduct. So this is for the capstones. So the end, we have the capstone course for the product development and process development. So we have two, because in engineering, we have only two, product development and process development. So to increase the knowledge about the economy, we also have the economic engineering also in the time. So maybe this is the subject in Faculty of Engineering related to the bio. For the biomedical engineering is maybe more special because they focuses on biomedical to the medical. So it's more to the medical compared with the general biotechnology or bioprocess. So they have also specific course for the like anatomy and so on. Maybe this is the answer for your question. Thank you.
Thank you, Dr. Sahlan. I hope we can we can accelerate our knowledge on bioengineering. It's really important from our countries. Any questions or comments? Maybe another questions from me, Dr. Salan. We are the same universities, actually, in Tokyo University of Agriculture and Technology. We BRIN have the nowadays is built good manufacture laboratories, in Cibinong Science Center, if I'm not wrong. Is it can be synergies together with bioprocess engineering in UI, and then we also have some position of a postdoctoral or visiting professor. Is it possible to work together with the facilities of BRIN?
Okay. As I mentioned in our slide, that we can collaborate in academic and also in research. Actually, maybe for the community engagement is also, but maybe I think it's not so much. But for the academic and for research, is very very good chance for us to collaborate. For example, in the case of the bioprocess, we already have the collaborations. I think with Dr. Puspita and others. I think BRIN have a good equipment, very good equipment. So why now we also have the collaboration in the case of our research about the rapid test. So our research is in BRIN, in this case in Serpong, in the Serpong. And I think it's also some of the analytical device, is very important. I think it's also, I don't know, for the postdoctoral maybe, postdoctoral. I think it might be our postgraduate students who's already finished their study. They can postdoc or after master maybe, after master. Is it possible for the master, graduate master to not postdoc maybe as a researcher to?
We accept only for the doctoral.
Maybe for the doctoral, because now we already have, even for the doctoral program, is still combined with the chemical engineering, chemical engineering and bioprocess engineering. So the students who want to learn about the chemical engineering in bioprocess stream, so they can also enter in doctoral program in chemical engineering. Maybe I think some of the students after finish can goes to the postdoctoral in BRIN. It is possible, and I think you have the good facilities also for scale up. The scale up is still difficult in Universitas Indonesia, especially for the scale up the productions and for the another assessment like clinical study and so on. So we need the CPOB, for example, or So I think I heard that BRIN have these facilities, and we can collaborate with this. And another one for the undergraduate, because we very strong in undergraduate. For the undergraduate, I think the joint supervision is the good chance, and also for the master course. we can collaborate, and because now it's full in our laboratory, so we can have to go to another laboratory. Yeah, thank you.
Yeah. Thank you very much, Dr. Sahlan. We got so many information, and then we can synergy together. As a short conclusion, the field of engineering should be learned and should be mastered by biotechnologists, microbiologists, biologists, zoologists, yeah, because by mastering the bioengineering, we can deliver our product from the lab to industry. Yeah, thank you very much, Dr. Sahlan. Applause to Dr. Sahlan. Thank you very much. Yeah.
Thank you very much, Dr. Puspita.
Yeah. Yeah. Ladies and gentlemen, next we move to plenary session. Today we have two plenary speakers. Our first plenary speaker today is Professor Eni Sudarmonowati. Let me to read her CV briefly. Prof. Eni is a research professor at the Research Center for Genetic Engineering, BRIN. Currently also, Prof. Eni is a visiting professor at the Department of Plant Biology, ISIBio, Rutgers University, USA, awarded fund from Fulbright. Also, Prof. Eni as a member of Species Survival Commission, IUCN, Associate Editor, Malaysian Applied Biology Journal. Prof. Eni obtained PhD from School of Biological Science, University of Bath, UK in 1991, and also bachelor degree from Bogor Agriculture University, Indonesia in 1985. Prof. Eni awarded Satya Lancana Wirakarya from the President of Republik Indonesia, Best Inventor, 100 Best Indonesian Women Researchers, Ministry of Women Empowerment and Children Protection, 113 Best Innovation in Indonesia, Ministry of Research and Technology, Ministry of Research and Technology. Her research interest in genetic improvement, genetic conservation, biodiversity assessment, genetic engineering, marker-assisted selection, cryopreservation, omics, and NGS. Professor Eni will be present the material for 20 minutes. After the second plenary speaker, there will be a question and answer session for 20 minutes. Yeah. Now allow me to welcome Prof. Eni Sudarmonowati with the topic of presentation, Genomic Approach for Modern Conservation and Genetic Improvement. Time is yours, Ibu.
Thank you, Dr. Puspita Listianti, as the moderator. Good. I think, can you hear my voice clearly?
Very clearly,
okay.
Yeah.
Yeah. So, good morning, good afternoon, good evening, wherever you are. Right now in here is, what's the time now? Is 9:41 p.m. in the US. So I'm very delighted to share my talk. Just let me share screen. I think it's here. Can you see my presentations? Slide presentation? Yes.
Yes, Ibu. Clear.
Yeah. Okay. Yeah. The title is Genomic Approach for Modern Conservation and Genetic Improvement. And the outline of my talk would be, as you see on the screen, I will, as the time given to me, only 20 minutes, I will be discussing a bit quickly, but those that you want to know further details, you can refer to my presentation file, because it's already downloaded in the link that already provided by the committee. So now I move on to the first one, development of genomic-based technology. We've been facing major threat, which is global threats, and then now becoming the world crisis. Five, the main one is habitat degradation, climate change, overexploitation and use of species, invasion of alien species and pollutions, which made us facing loss of biodiversity. And also a number of animal, plant and fungi species are now listed as threatened by IUCN Red List, and most known species, like 72%, still need to be assessed. Before move on to the what the genomic can do for the conservation, I would like to share with you the progress of the development of the genomic technology since 1966, and then move on to microsatellites in 1990, and then 2000 SNPs. Everybody now use SNPs, and then for population genomics, it's more on 2010. I will be discussing more on this later on. But in here, you can see detection of inbreeding depression that it's difficult to detect using the conventional conservation technology. Just to refresh you, we have 3 sequencing generations. This is just to refresh. The first ones established in introduced in 1977 with medium read length, still low throughput, and then still high cost base. And then the second one between 2005 and 2010, already high throughput, drop the cost already. And then the third one between 2010 and 2014, it's already long read, and the price, the cost even dropped, becoming more cheaper. But then the development still go on with the establishment of NGS. There are some advantages of NGS. You can rate it here a more lower cost. And then they use a maximum number of SNPs, and also the development for genomic assisted breeding, the GWAS, the Genome Wide Association, using the utilizing the next generation sequencing, and also the GPS genotyping based sequencing, then develop a QTL map, genomic selections, and then the modification, genetic modification using CRISPR-Cas9 after the transgenic era. Just to give you a very brief information on the genomic information already available, already sequenced, and you can find it in the NCBI gene bank. You can see here all different organisms, like the most is Animalia, the green one, the most information available, and then which one is the complete sequence already, the one with the red color. So the choice is yours. I mean, depend on the objective that you targeted. So either you're going to use the whole genome sequence or the whole genome resequencing. So it really depends on the target. But the whole genome resequencing offer you the possibility to screen a neutral and functional polymorphism in high genomic resolution. And further development and moving toward functional genomic, I listed the advantages here, 7 advantages, but there are more. And here, including the wildlife and humans, and also the tolerance and quality traits assessment, and also more powerful technology or mechanism for signaling pathway. And now I would like to move on the genomic roles in conservation of endangered species. And here, why there are some endangered species difficult to be conserved, because there are some obstacles like limited mating pairs, inbreeding, and also inbreeding depressions, so that we need to evaluate which species is vulnerable or less adaptability to the local conditions. So that's why we need to use the suitable technology to address this one. So in this chart, you will see this one is more on the conservation flow chart, but the one with the red one, that actually can be addressed wider when we use genomic as compared to conservation genomics, conservation tools. And the one in green, like genetic drift, inbreeding, hybridizations, can be more precisely estimated by genomic technology. And in here, I just listed several advantages of what genomic can do for conservation, genetic conservation, such as precise genomic monitoring of changes in allelic frequency, and then quantifying the effects of genetic drift and natural selection, hybridizations, and so on. And the advantages of new approaches, not dependent on genomic information already available from the closely related species, so you can do from scratch. So for the conservations, I mentioned earlier that some endangered species are difficult to be conserved. So they declined, the population in the wild declined very rapidly. So then we need genomic approach, genomic-based approach. I mentioned some of them already earlier, but here then because it provides the potential to understand the genetic basis of interaction among species, and also genomic monitoring of changes in allelic frequency to quantify the effects of genetic drift, for example. I listed here up to 20 of the advantages to use genomics for conservation. I don't want to read all of them because of the time, but just to mention, among others, it's actually like the adaptation to local conditions, demographic history, and historical effects, size. Oh, sorry, I listed 2 in here, twice, double. And then the phylogenomics and evolutionary history, population substructure, population connectivity, and so on. And this one, because it provides, this slide shows you, because it provides quite straightforward contributions, such as addressing some problems and then offering possible solutions through genomic approaches, such as minimizing adaptation to captivity, predicting the viability of local populations. And also it offers, with all of our roles, of course, how to slow down the bend the curve of biodiversity loss. So in here, this curve, it's actually used by UN, United Nations, on the meeting of CBD. So the down, the bottom, the gray one, the gray line, if you just do business as usual. You do nothing. And then the yellow one, increase conservation efforts, so it will bend a little bit of the curve. But the green one, if you do increase conservation effort plus more sustainable production and more sustainable consumption, then it will bend the curve of biodiversity loss. Among others, the technology is using the genomic approach because I mentioned earlier that we can prevent genetic pollution and also evaluate its conservation values and so on, and also the migration rate. So what a genomic can do for genetic improvement, because most characters actually regulated by various genes, multiple genes, complex traits, so that it's difficult and take a long time to use this conventional breeding. So using the genomic-based approach, then with the technology I mentioned earlier, the next generation sequencing, among others, and also other technology, then combining with others, omics and also gene editing system, then it can fasten the way to get the targeted one. So I now move on to research and applications on conservation. So I show here the genomic-enabled discoveries and application on humans, livestock and wildlife, plants, insects, fungi, bacteria, and viruses. For example, for microbiome in health and disease, for fungi, for synthetic biology, and others for plants, for genomic selections, also for livestock, and also the diagnostic for human. This one, sorry, this one not very clear, but in here is the example in wildlife and animal, and also what the aspect also already done, already been done. For example, on wolves, monkey, birds, and also some plants also. This is just a summary. And in fish, in Amazon, in Latin America, this one, the symbol fish are Paima gigas. The genomic approach can already offer the fishing management and also genetic management for aquaculture, so that this fish that are almost extinct in the wild then can be increase the population in the wild also. And also it provides low-cost approaches and low-cost infrastructure. For Pacific salmon, this one just to give you several examples, also can give you the migration phenotypes, so that the management, conservation management can be adapted or applied using SNPs. And for Tasmanian devil, in Tasmania, this one almost extinct in the wild, but using genomic approach, the GWAS and also the SNPs, then can be prevented from extinctions, and now they can be, it raised the population. And in bird, also in this one, the endemic and also threatened birds in New Zealand. So now using the whole genome resequencing and SNP, so they can produce this one, the conservation, the population also increase in the wild. I mentioned earlier about the adaptability and vulnerability of certain species. So this one, I just give you the case in Japan that especially in small islands. So this one using the assessment with the whole genome sequence and also the next generation sequencing, then they can provide a recommendation for the government how to preserve this one. And also another plants on rhododendrons, also they provide the government to give effective conservation strategy. Because I'm now in the US at Rutgers University, so I know I also witness the research on this one on conservation, but as well as also for breeding of cranberry. It's famous for sourness, but it's has abundant health promoting compounds. So using a bulk segregation approach with SSR and QTL, and also the GBS, the genotyping based sequencing, so they can produce they can conserve and also produce improved materials of cranberry. For woody species, sorry, not many people working on this because it's a long life cycle, but I just listed the one in Indonesia just to give example, but there are many abroad, internationally, genetic diversity, population genetic assessment and genome sequence of certain species of Indonesian timber, such as the ironwood, the Aeschylodirexulan swageri, and also the woody species like Mangifera casturi. This is already almost extinct, but now can be propagated, and also the assessment already been done. And transgenesis and the genetic stability test. So this one, the impact of logging and the impact of forest fragmentation already analyzed, assessed using genetic markers in the past, but still the recommendations being used by the government that we need to increase the population in the wild. And this one on Ratan Manau, and also other species under Diplocarp. And now move on to the research and application on genetic improvement. So combining between gamma irradiation and microsatellite markers, two species of Acacia mangium and Paracentus vacacharia can be produced, improved one, the mutant one, the straightness of the stem and the timber, and also the permanent branch, and also the height and also the height and the branch from the surface, the branch height. And to confront with SSR, and then this one using ISSR on cassava to several traits, related to several traits, and also the SNP for Psi gene regulating beta carotene also already been done. And also detecting the transgenic plant, this one trees on the less lignin content and high cellulase already been expressed. And this one on at least four or three species in Indonesia. One of them is endemic Indonesia, the Paracentus vacacharia. And the overexpression of the genes stable after 8 years. We tested and also planted. They're still able to regenerate and still stable, confirmed with the molecular analysis. So since again, I'm at Rutgers University, I came with them, this Professor Jim Simon and Dr. Rong Di produced gene-edited basil. Basil is Indonesia like kemangi, resistant to downy mildew disease. I came with them to the experiment station there. So here, you can see the improved basil, very fresh and green. At the back, the one that I put the green light or green pant or something, that's the white type. It's already devastated by the downy mildew disease. So it's quite significant, the effect of the gene CRISPR gene edited basil. So this one using the gene homoserine kinase. And for rice, for sweet potato, for wheat, this one from USDA also in US. So they use a different kind of technology like GWAS, and then also other markers for the traits of grain dimension, cooking quality, beta carotene, and others like drought tolerance. So it's really depend on your target And also this one for fruit species, durian, volatile sulfur compounds, and papaya for the fruit weight, fruit length, and so on. Banana for the carotenoid rich, and other fruit for the shape and juiciness. And I listed here also the progress on functional genomics on wheat. So for tolerance and most for drought tolerance, combining transcriptomic, proteomic, and metabolomic using SNP and rapid markers. And now move on the recent and ongoing collaborations between Indonesia and in this one BRIN and Kyoto University and Tohoku University on here you can see the status endangered, critical endangered, and mostly critically endangered and endangered of 4 species using whole next generation sequencing. And also this one is just I just skip this one one of the detail of the species.
2 minutes more.
And and then now using the white durian, this is the first one. We will be the first draft genome sequence of the white durian. So this one, durian cutiensis and durian oxyanus. And then we will generate the PSMC to detect the demography structure population in the past, like 1,000 years back, and also in the future. This one a new technology as compared to PSMC, but also the MSMC, and then this one another new approach using star wave plots. Of course, there are plus and weakness, but this one does not require whole genome sequence as a reference genome. And then we're going to do another 2 white durian for producing draft genome for the first time also in the world, so hopefully it will be soon. And then for networking and collaborations, we need the all participation of all of us, governments, NGO, funding agency. And these are all different type of collaborations, bilateral, regional, global, and others. So we need to make use of that. I just give you the example in the past, a bilateral collaboration with Indonesia, Netherlands, Indonesia, UK, Indonesia, Zurich. So this one on cassava, on stock compositions, and also for extending the shelf life of cassava. And lastly, before conclusions, this one, what the research result for if is it not used by our users? So this one of the users is the government. So we need to give the recommendation for the conservation. So in these 2 books, there are also genomic approaches, the SSR and other markers that have been assessed, used to assess the conservation. And to conclude, lastly, the genomic approach provides more efficient, this one reduce time and low cost for human wellness and nature, not just human wellness, but also nature. So on the first day, yesterday, you all heard from Dr. Lily, the BCG. So this one, bioeconomy, circular economy, and green economy. And the second one, for this technology, we can provide recommendation for national conservation strategy. We can bridging the gap for breeding of elite clones or improved strains or for microorganisms and others. And also we still have a lot of homework to do because certain technology need increased or robust regeneration protocol for many species, and research need to be adopted. So that's our efforts need to be increased for that. So thank you very much for your attention. So I would be very happy to have your questions. Thank you.
Thank you very much, Prof. Annie, for introduce us your work until today. The next of plenary session is Associate Professor Dr. Insinyur Hasimi Abu Hasan Is it already here? Yes.
Assalamualaikum. Can you hear my
Waalaikumsalam warahmatullahi wabarakatuh. Yes, I can hear
voice
clearly.
clear?
Yes, very
Clear.
clear.
Okay.
Yeah.
Okay. Very much. I
Yeah.
try to
Associate Professor Dr. Insinyur Hasimi Abu Hasan is a head researcher at the Research Center for Sustainable Process Technology, University Kebangsaan Malaysia. And then also associate professor at the Department of Chemical Process Engineering, UKM, and also associate research fellow, Research Center and Water Analysis, Faculty of Science and Technology, UKM, managing committee member, International Water Association, and then obtained PhD from University Kebangsaan Malaysia. His honors and awards obtained was Anugerah Bintara Ahli Akademik Harapan UKM, Most Valued Reviewer for Bioresource Technology Elsevier, Anugerah Bintara Ahli Akademik Harapan also from UKM. The research interest is on a waste treatment, waste audit, resource recovery, biofilm, phytoremediation, and remediation. Professor Hasimi Abu Hasan, time is yours. You will make your presentation by yourself?
yes, sure.
Okay.
Thank you, Mr.
Thank you very much.
Chairperson. Okay. I will share my screen. You can see my screen, right?
Yes,
So
we can see yours.
Yes, okay. 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 Thank you for inviting me to give a speech in this conference. Okay. As presented by the Mr. Chairperson, my name is Hasimi Abu Hasan from Department of Chemical and Process Engineering, and acting as head for the Research Center Sustainable Process Technology. Okay, so my topic today is on the how to treat the river or how to treat the surface water through the biological approach. Okay, just a moment. I try to remove this one. How to? Okay, never mind. Okay. So so this is my outline for the presentation for the speech. Okay, I will start with some introduction on the environment, especially on the river. Okay, and then what kind of the river pollution in the in especially in Malaysian country. Okay, and then the treatment technologies we use to treat the river. Okay, to produce clean water, and then the challenge. Okay, the challenge to use the biological approach. Okay, and the last one, of course, we have conclusion. Okay, as introduction, as a human, we need a water. Okay, because water has an important role in our metabolic process. Okay, so this water it will break down a large substance into a small substance. Okay, so what happens if the water contaminated by the pollutant? Okay, of course it will affect to our body. Okay, So the contaminated water will become hazardous, okay, not only to human as well as to plant, hazardous to the animal, and this kind of pollutant will expose through the drinking water. Okay, so there are a lot of pollution sources in the river. Okay, for example, from the industrial discharge, okay, the pollution from the illegal garbage. This is the common one, pollution from the illegal garbage. Okay, and then pollution from the sewage effluent discharged to the river without proper treatment, and then from the leachate, okay, leachate landfill, and of course from the agriculture industry. Okay, so agriculture industry can pollute the river through the water runoff, okay, during the watering or during the raining season. Okay. Okay, this is some of the river water pollution. Okay, in terms of chemical compound. Okay, there are 4 categorized here. Okay, the first one is heavy metal. Okay, there are many heavy metal contamination in the river. Okay, for example, plumber, cadmium, or zinc. And then other parameter is inorganic, non-metallic. Okay, so inorganic metallic, the source for this kind of pollution is from the sewage industry, from the industrial industry. Okay, and for example, of the inorganic non-metallic pollutants, nitrogen compounds, sulfate, or chloride. Okay, and then what else? The river water pollution. Okay, endocrine disrupting chemical, EDC. Okay, and disinfection byproduct. Okay, these 2 categories are under micropollutant types. Okay, we could not see by our naked eye. We can analyze both categories using high-end equipment, and not very easy to analyze. So this is the current issue in the river, okay, around the world. Okay, so and then other kind of the water pollution are physical. Okay, so the physical contamination contribute by the garbage. Okay, and then once we throw the garbage into the river, so it will contaminate the river, of course, as well as the color. It will create the bad color, the black color, the brown color of the river. And then we can see the turbidity, the clearness of the river become reduced, okay, through the turbidity measurement. Okay, this is some example, the water pollution in Malaysia. Okay, some news, okay, about the contamination. Okay, and that news in Malay, I think you can understand. Okay, you can see the fish in the river. Okay, died because of the pollutant. The river water become toxic. Okay, and not only die, some aquatic plants also become yellowish, start to decay. Okay, and some other example. Okay, in Malaysia, there are many. Each year we can hear about the water contamination, not only in Malaysia, also in other countries, especially in developing countries. Okay, this is some other example. So you can see this in English. The river water turn green. Okay, this is because of the dye discharge. Okay, from the textile industry. So they discharge the dye directly to the river without any proper treatment. Okay, so in Malaysia, we have a conventional treatment, okay, to treat the river water. if the river are very contaminated at high level, of course, the conventional treatment could not be operated for the production of clean water. Okay, so in Malaysia we have the treatment of the grid chamber. Okay, this one, and bench stream. This is to remove the garbage. Okay, or to remove the large particle. Okay, for example, the dead leaf, the trunk of the plant. Okay, and then we have the aerator here. Okay, aerator is purposely to reduce the light heavy metal, for example, the ferrum, the manganese. Okay, and then followed by the flocculation and sedimentation tank. Okay, this is to remove the dissolved organic material. Okay, for example, to reduce the suspended solid, to reduce the dissolved metal, so we use the flocculation process. And then we have a sand filter. Okay, before the end of the treatment. Okay, sand filter to remove residual suspended solid. And the last process, we treat the water by the chlorination. This is to ensure there is no microorganism contamination in the water. Okay, so our purpose, okay, why we develop a technology to treat the river water using the biological approach is in the current technology we could not remove the nitrogenous contamination. For example, we could not remove the current technology could not remove the ammonia, could not remove the nitrate, could not remove the nitrate. So by having the biological approach, okay, so those pollutants could be removed effectively, and at the same time the water treatment in Malaysia continuously operated, okay, without any shutdown. So this is what we propose and what we study in Malaysia in my department. Okay, biological treatment is one of the advanced treatment. Okay, so it started popular since 1900, but not for the water treatment. It's popular for the wastewater treatment. Okay, so the application is still limited around the world. Okay, but for the developed country such as US, Canada, they have very well knowledge on the technology for the drinking water production or for the river treatment. Okay, so of course, when we're talking about the biological approach, we have to make sure that the process use non-pathogenic bacteria. Okay, so this kind of bacteria will act as biocatalyst for the biochemical oxidation, and then it can degrade the pollutant in the river. So the technology could be proposed before the chlorination process. Okay, because if you propose at the end of the process, it might be contaminated with the microorganism. But if you propose before the chlorination process, so the microorganism contamination could be avoided. Okay, so now we go to the what kind of the technology for the biological approach. Okay, so we can use the slow and rapid sand filtration. Okay, so we have published a few articles on this technology, and this technology could remove the light heavy metal. For example, it could remove the manganese, it could remove the ferrum, it also could remove the plumbum, okay, in the river water. Okay, so but the lacking of this technology, it could not be operated if the heavy metal too high in the river. Okay, too high means it could not exceed 2 ppm. Okay, if the concentration exceeds 2 ppm, so we could not use this technology. Okay, because the heavy metal could provide a toxicity to the microorganism. Okay, so this is some explanation about the sand filtration. Okay, there are two types of sand filtration. The first one we call slow sand filtration, and second one is rapid sand filtration. Okay, so this process used normally we used to filter out the suspended solid and light heavy metal. Okay, so the sand filter, okay, that fail in the reactor, okay, will act as a support for the biofilm growth. Okay, once the biofilm grows, it can provide a space for the biological activity. Okay, and we can design the sand filtration in a column shape or in a rectangular shape. Okay, this is the biofilm layer on the sand surface. This is in German language. Okay, we call it smart Dutch. Okay, and this biofilm layer contain extra polymeric substance. Okay, so this substance could react with the pollutant through the bioaccumulation, through the bioabsorption, and of course we can produce the clean water. And this is some of the performance for the biosand filter to clean the river. Okay, especially the ferro and manganese as a light heavy metal, as well as the ammonia. Okay, we can also treat the ammonia if the ammonia level high, more than 3 ppm, so we can use the sand biofilter. Okay, next is biological activated carbon. Okay,
you have 5 minutes more, Sensei. Yeah.
Okay, I will speed up my presentation. Next is biological activated carbon. So this kind of technology use the activated carbon to absorb the pollutant. Okay, so there are two mechanisms for the biological activated carbon. The first one is aerobic process, and the second one anaerobic process. So the activated carbon or the adsorbent could be regenerated through the microorganism role. Okay, so the microorganism will consume the pollutant, and at the same time the pollutant from the activated carbon will consume back by the microorganism for their growth. Okay, this is the performance. It can reduce the micropollutant, okay, something like EDC. Okay, and then trickling filter, another treatment. Okay, this is not a common technology for the trickling filter, but we can use the trickling filter to remove the manganese, okay, iron, and also ammonia. Of course, this technology will involve the microorganism, something like manganese oxidizing bacteria, ammonia oxidizing bacteria, for example, Nitrosomonas, Nitrobacter. Okay, and also we can use the biological aerated filter. We have published more article for this kind of technology. Okay, this will involve the using of floating carrier. Okay, the floating carrier will act as a house for the bacteria to grow, to attach, and at the same time it can provide a large surface area for the biofilm, for the contaminant removal. Okay, this is the performance. And then we also could use membrane bioreactor. This technology combination of the bacteria, especially bacteria, and the membrane. Okay, and there are two type membrane technology. Okay, we call it as attached growth membrane bioreactor, and we can also set as ion exchange membrane bioreactor. Okay, so normally we use the membrane bioreactor at the end of the process before the chlorination. Okay, And also we can use the moving bed by film reactor. Okay, this technology combining the activated sludge process and also with the aeration, we provide some aeration for the bacteria. Okay, and the aeration could create the dissolved oxygen concentration in the contaminated water, okay, normally at 2 until 5 ppm. Okay, so there are also some integrated biological approach. Okay, with other technology. Okay, for example, we integrate the membrane. Okay, membrane bioreactor with the coagulation process. We add some chemical before the treatment. Okay, so the chemical will reduce the suspended solid or will reduce the light heavy metal, and the treated water will go to the membrane and it can increase more the pollutant removal. Okay, and then we also integrate with the adsorption technique. Okay, between the adsorption and the membrane. Okay, we call it as biological activated carbon membrane bioreactor. Okay, so this is the combination. And then what is the challenge in using biological for the drinking water treatment? So we have to face, we have to solve how to make sure the treated water are free from the pathogenic microorganism contamination. Okay, this is the challenge. We should have a frequent monitoring on this parameter. And then the selectivity of the microorganism towards the pollutants. Okay, not all microorganisms have capability to remove the pollutant. So we should provide the system with a good microorganism that could remove what parameter we need. Okay, and then the third challenge is to maintain the growth of the microorganism in the system. Okay, this is very challenging, and of course, once we produce the drinking water or we treat the river, what is the consumer perspective? Okay, are they worried about the contamination of the microorganism or not? Okay, so we have to do some survey for this. Okay. And at the end, for the conclusion, of course, this is a new technology or our future technology for the river water contamination, as well as especially for the developing country. And there are many area that we can explore. And of course, this technology could be growth well in developing country. Okay, thank you. I think that's all my presentation. Before I end, I want to say something. The earth is beautiful. Let us maintain the beauty by caring for the environment. Okay. Okay. Thank you very much, Mr. Chairperson.
Thank you very much, Dr. Hasime Abu Hassan. Ladies and gentlemen, now are the time of Q&A session. Please using chat box, chat feature of the Zoom and raise your hands, and then from whom the questions will be delivered. We have 10 minutes for the question and answer session. And then there is one question from the chat book to Prof. Annie. According to your experience, what method is the most effective and efficient in transferring the desirable gene into the plant? From Ibu Elok, BRIN.
Okay, thank you for the questions, Ibu Elok. Yeah, so nowadays, because of the development of CRISPR-Cas9, this is the most effective way. I mean, efficient, because efficient when we talk about the resources. So it's quicker in terms of time. I mentioned in my slides, I showed to you all the successful research using CRISPR-Cas9 on basil at Rutgers University in the US. So this one, they only needed 2 years to get the gene-edited plants as compared to the one that using the conventional breeder, they need 7 years. And then how to deliver the gene after it's cut by CRISPR-Cas9. For me, for my experience also, and also when I'm here at Rutgers, using particle gun as compared to agrobacterium tumefaciens. Because when you use agrobacterium, it's almost most of the time you will be like deleting or to get rid of the bacterium from the plants. I mean, if the concentration is too high, then your time is consumed for transferring or get rid of the agrobacterium. But using particle gun bombardment is better, because then you don't have to be disturbed with the agrobacterium. So in here at Rutgers University also, they use the particle gun bombardment of Bioresources. I mean, last time I used not Bioresources because we used the cheaper one, but less effective. So I think we need to have the Bioresources also. And also, although either you use the conventional genetic engineering or using the transgenesis or CRISPR-Cas9, again, as I mentioned in my conclusions, one of the list that the regeneration procedures is need to be established. So no matter how high or how advanced is your technology, if you want to improve genetically, you have to establish the regeneration system. Yeah, so that's my answer. I hope that's satisfy you. Thanks.
Okay. Thank you very much, Prof. Annie. And then from the chat book to Prof. Hasimi from Ibu Penny Lestari. The result of this research are very good and protecting the earth from contaminants. Is the use of bacteria of microbes to treat pollutant also safe for human? Or how to manage these microbes? How far have the results of this research progressed? Has it been applied in product form? How to get it?
Okay.
Please, Dr. Hasimi. Yeah.
Okay, thank you for the question. Okay, apologize. Okay, there are few questions, right? Okay, how to manage this microbe? Okay, during the treatment, okay, we could monitor the growth of the microbe, okay, through the what we call it, mixed liquor volatile suspended solid, okay. Or we can measure through the adenine triphosphate analysis, okay, ATP. So this value represent the growth of the microorganism. Okay, once we produce the treated water, okay, after the biological treatment, we will treat with the chlorination. Okay, so the chlorination will destroy all the microorganism, and we will make sure after the chlorination process, there is no microbe contamination in the water. Okay, this is what we have done. Okay, and then so of course the safety of the treated water to the human. Okay, so this is the challenge that we have faced. Okay, so the consumer will worried to consume this water because they think it contaminated with the microorganism. Okay, so we have to convince the consumer or the human that the water is safe. Okay, because we are using the non-pathogenic bacteria. Okay, not pathogenic bacteria. Okay, so and we also control with the chlorination. Okay, before we distribute the water to the human. So through this control, okay, so we can ensure that the water are safe to the human. Okay, so and then so far this project is still under research progress. Okay, not apply in a real scale or pilot scale. Okay, of course, there are more study to be explored. Okay, especially the consumer perspective. Okay, and there are one question about the economic. Okay, so for the economic test or analysis, this still ongoing. Okay, but at the lab scale, not at the pilot or real scale. Okay.
Yeah.
That's my answer.
Okay, and I want more answer. How about the study from economic side when it will be applied?
Yeah, of course, in terms of economic side, okay, from the review, okay, this technology should be more cheaper compared to other chemical technology. Okay, because we use less chemical. Okay, and then it's easy to operate. Okay, easy to operate. That doesn't mean we do not need a competent staff or competent operator to operate. Okay, still need the competent operator, but easy to operate, easy to learn. Okay, just to competent how to manage the microorganism growth in the system, and of course, economic side is more cheaper compared to other technology. Okay.
Okay, thank you very much, Dr. Hasimi. We will be finished for this plenary session. Thank you very much, Prof.
Yes,
Annie.
there are two. Sorry, there are two.
Oh, yeah.
There are two raising hand.
Oh, I'm sorry. Oh, yes.
Yeah.
Pak Panji and Ibu Yeni. Please, first, Pak Panji.
Do I still have time, by the way?
Yes, we still have 1 and 2 minutes.
All right.
Okay.
All right. I'm just going to make it quick. I have one question for Bu Eni and one question for Pak Hasimi. For Bu Eni, I am studying microbiome now, and I learned that the loss of the microbiome would influence the loss of the high organisms as well. Do you think that targeting the microbiome in addition to the genetic identity of the microorganism would help to preserve the endangered species? And for Pak Hasimi, I am now doing a project with KU Leuven about the use of microbial inoculant and as well as the horizontal gene transfer in microbiome to reduce the pollutant. And one of the key challenges that we face is to maintain the survivability of the microbial inoculant. How did you maintain yours in your system? Thank you very much.
Thank you very much. Please give
Can
the
I
short
answer?
answer.
Yeah, very short one. Okay, before I also short because of the time. Yes, definitely studying the microbiome will affect the degrading or increasing the populations or enhance the loss of biodiversity or maybe improve the population size. That's why the study in microbiome is very important, and also because it's also related to the gut, even in the intestine of the animal or wildlife or even human. So that need to be studied further. Yeah, so that's why the study in microbiome is very important, because it's related to the extinctions or the survival of the species. Yeah, that's my quick answer.
Ibu Eni. Dr. Hasimi, please. Yeah.
Okay, how to maintain the growth of the microbe in the reactor. Okay, what we do is, the first step is we have to make sure the incoming river water to the system is not at very high contamination. Okay, so we should remember this technology is used not at the first stage. We could use at the second stage or the third stage. Okay, if the concentration of the contaminant is very high, so it will kill or will become toxic to the microorganism. Okay, the contamination depends on what parameter we want to treat. Okay, and for example, if we want to treat the ammonia, okay, we have to make sure the ammonia level after the first stage of treatment, it should be not more than 3 ppm. Okay, so we are talking about the water, so the contamination should be not high, and from that it will maintain the growth of the microorganism, and we can monitor the growth through the mixed liquor, volatile as well as solid. Okay, that is what we have done in our research.
So by that you mean you get the microbiome to adapt to the environmental stressor that would...
Yes.
Okay.
Yes,
right.
we have to do that. Okay, to make sure it can adapt with the environment, it can have a good removal stability performance.
All right. Okay. Thank you very much, both.
Okay.
Thank you very much. I'm sorry, Ibu Yeni, the time is up. And then thank you so much again, Prof. Eni, and then Dr. Hasimi, for the very informative and then inspiring presentation. Yeah, I move this session to MC.
Thank you.
Yeah, thank you so much.
Thank you.
Thank you, Prof. Eni. Thank you, Dr. Hasimi, and also thank you, Dr. Puspitalis Dianti, as the moderator of the sessions. Ladies and gentlemen, following to the next agenda, we will have the parallel sessions consisting of 5 rooms with different topics, and also each of room will be led by moderators. So the first room will be bioresources and biodiversity. The second room will be nutraceuticals and natural products. The room 3 will be biotechnology and bioinformatics. Room 4 will be medical sciences, and the last room, biorefineries and circular economy. So please kindly choose the room based on your interest by clicking the breakout room feature at your Zoom. If you haven't found it yet, you can click more with 3 dots at the corner of the left of your Zoom meeting, then join breakout room there. The room 1 until 5 are available there. Once again, please kindly choose the breakout room by clicking feature more and then join breakout room.
Afternoon. I'm sorry, committee, can we directly start or is there any, or we need to wait first?
Sorry, Tina,
Yes.
increase the volume. Okay.
Can you hear my voice?
Okay.
Is it okay?
Okay, it's clear.
Should we directly proceed to the presentation or we need to wait until 10:50? Can you hear my voice?
Yeah, I think we can wait until 10:50. And
Also,
then,
so we wait until 10:50, right?
yes,
Okay, then we will wait for 3 minutes more. Okay, good afternoon, our distinguished presenters.
Recording in progress.
Welcome to the today conference ISIBio 2022. So before we would start our conference today, I would like to introduce myself. So my name is Vina Laksmi. I would be the moderator for this session. So this is room 1 with the topic bioresource and biodiversity. And today I would like to introduce our 4 distinguished presenters. There is Miss Carla Frieda Panto. And then second presenter is Dini Hari Indah Kusumawati. And the third presenter is Septiantina Diahriendria Sari. And the fourth presenter is Aida Ulansari. So before I start the presentation, I would like to check if the distinguished presenter already here. So Miss Carla,
Yes, I'm here.
Okay, thank you very much. And then next is Miss Dini Hari. Are you there?
Yes, I'm here.
Okay, thank you. And then our third presenter, Miss Septianti Nadia Riendriasari.
Yeah, I'm here.
Okay, thank you. And our last presenter will be Miss Aida Wulansari.
Yes, I'm here.
Okay, thank you. So our four distinguished presenter already here. I would like to start the presentation today with the first presenter, which will be Miss Carla Frida Panto. She will be presented his research result with title Plant Biodiversity in Kapuas Hulu West Borneo, Study Case of Danau Sentarum National Park. So time is yours.
Welcome to ISIBio 2022. Our group are going to present our research in section with the title Plant Diversity in Kapuas Hulu, West Kalimantan, Study Case of Danau Sentarum National Park. Our group member Sahal Muaz and Carla Frida Pantau are represented by me, Bernadetta Rina Hasti Lestari. One of the keywords in this research is the National Park. Petung Kerihun and Danau Sentarum National Park, or abbreviated as NBKDS, is located in Kapuas Hulu, West Borneo, and this area is very unique as it belongs to transboundary conservation area between Indonesia and Malaysia. The total area of this national park is around 800,000 hectares, differentiated as two parts. The first part is Petung Kerihun National Park, which ecosystem is mainly terrestrial, and Danau Sentarum, which ecosystem is mainly aquatic. This area is rich of tropical biodiversity, as well as culture and local wisdom. This national park has been designated as UNESCO World Biosphere Reserve on July 25, 2018. Focus of our research is in Danau Sentarum National Park. This national park occupies more than 130,000 hectares of the floodplain of the Upper Kapuas River. This area covers seasonal open water like lake, swamp forest, and some lowland forests on the hills. This area will appear differently in the rainy season like this, and in the dry season like this, with low volume of water in dry season. The area becomes a natural dam for supplying fresh water in the dry months and preventing flooding in the rainy months. Besides that, this area is a habitat for various flora and fauna, as well as local community livelihood. The community living in this national park depend on dry farming with additional activities, for instance, fishing, hunting, poultry, and forest resource collection, as well as gold mining. As the number of national park staff is limited, the identification of vegetation in this park is not optimal. Therefore, we are going to do research with the objective to study the plant diversity in Danau Sentarum National Park, Kapuas Hulu, West Kalimantan. The method. This area covers 7 sub-districts that are Badau, Batang Lumpur, Bunut Hulu, Lembau, Selimbau, Semitau, Suait. And these sub-districts are managed by 3 management office, that is Lanjak, Semitau, and Selimbau. This large area is differentiated into several zones that are core, jungle, utilization, traditional, rehabilitation, and special zones. Our research in a traditional zone because it's the representative of the majority zone and it's near the Lanjak management office. The sampling was conducted for 1 week on 19 to 24 June 2022. A drying plan for herbarium specimen was made using the method of Jarwaningsi et al. 2002, and identification of morphological characters, direct and laboratory observation of vegetative and generative structure were also conducted. This area could be differentiated as peripheral and middle area. The peripheral area was dominated by tall trees of family Dipterocarpaceae. This family has characteristic of single, alternate, thick leaves, whitish brown to brown stem, and deep taproots. The identified vegetation can be seen in this table, with status varies from least concern to endangered, and also critically endangered. The endangered species was Cotyledon burki and Sorea placipilla, and the critically endangered is Sorea platycarpa. The vegetation around the middle area of Danau Sentarum National Park can be also seen in this table. In this table, it can be seen that the status of scarcity varies from least concern to near threatened and vulnerable. Some vulnerable species are Nepenthes bicalcarata and Tetramerista glabra, and the near threatened species is Placonella maigani. Here are some pictures of the identified vegetation. First is the Nepenthes bicalcarata here in the habitat, and the second is Nepenthes bicalcarata when we look closer, and the Sena alata and also Floyarum alternifolium can be seen mostly in the middle of the area. And the example of the Dipterocarp species is Sorea platycarpa. The conclusion of our research is that vegetation of Danau Sentarum National Park is dominated by Dipterocarp species with status of scarcity is concerned to critically endangered. Assessment of vegetation is crucial for the sustainable conservation. Regeneration of the species, ex situ conservation, and overexploitation prevention are crucial to anticipate extinction of species. We would like to acknowledge the Expedition and Exploration Funding Batch 1 Directorate of Research and Innovation Facility for funding this field work. We would like to thank you, Research Center for Genetic Engineering National Research and Innovation Agency, the NBK team, Melia Community, Marangat Foundation, and you for your attention. Thank you.
Okay, thank you very much for our first distinguished presenter. So before we would proceed to our second presenter, I would like to inform all of you participants here that you could convey your question in the Q&A session after all of the presenter here finish their presentation. So we can proceed to our second distinguished presenters. Wait a minute. The second presenter is Miss Dinihari Indah Kusumawati with the title of her presentation is Diversity of Cellulose Degrading Bacteria from Imperata cylindrica grassland in Indonesia. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Distinguished guests, ladies and gentlemen. Today I would like to share my research result with the title Distribution of Cellulose Degrading Bacteria from Imperata cylindrica grassland in Indonesia. This work is a part of SATE project. Alang-alang or Imperata cylindrica is one of the species that widely distributed over tropical and subtropical region. The disadvantage of alang-alang is causing problem in agricultural production. On the other hand, the advantage of alang-alang is can be used as raw material for bioenergy and biofuel due to fast growing, relatively easy to harvest and process, less land maintenance, and it can be an essential contribution to the reduction of anthropogenic carbon dioxide. From our previous study, we successful isolation of solubilized of GA phosphate complex and release of phosphate bacteria, nitrogen fixing bacteria, inoacetic acid oxidizing bacteria, and cellulitic degrading bacteria from 3 locations of Imperata grassland in Indonesia. This study aims to investigate the distribution of 18 cellulitic bacteria from 3 locations by identifying based on molecular technique. 18 cellulitic degrading bacteria obtained from the previous study were used. 7 strands isolated from grassland soil from Malacca, East Nusa Tenggara, 6 strands isolated from grassland soil from Katingan, Central Kalimantan, 5 strands isolated from grassland soil from Cibinong, West Java. All bacteria were isolated by direct and enrichment method using selective agar media to obtain specific genera of VGPR, and those isolates were screened for their ability to degrade cellulose qualitatively. Molecular identification and construct phylogenetic tree based on molecular method determined for all isolates. As a result, 18 cellulitic degrading bacteria belong to the 4 phyla and 10 genera, which is phylum Pseudomonadata, Secundra, phylum Bacillota, 2 genera, phylum Bacteridota, 1 genus, and phylum Actinomycetota, 1 genus are found as shown in this video. Phyla Pseudomonadata and Bacteridota distributed in Malacca, Katingan, and Cibinong soil sample. Phyla Bacillota and Actinomycetota only distributed in Malacca grassland sample. In 2021 report, a killer phyla named self-nomenclature of prokaryote. Pseudomonadata is a later synonym of Proteobacteria, Actinomycetota is another synonym of Actinobacteria, Bacillota is synonym of Firmicutes, and Bacteridota is a synonym of Bacteroidetes. Distribution of the bacteria at 3 Imperata grassland based on the genera is shown in this slide. RM soil sample from Malacca, East Nusa Tenggara consists of bacteria from 4 phyla, Pseudomonadata, Bacteridota, Bacillota, and Actinomycetota, with 7 different bacteria genera: Enterobacter, Pedobacter, Bacillus, Acrospinobium, Mycobacterium, Neisseria, and Myxobacter. However, KT soil sample from Katingan, East Kalimantan consists of 2 phyla, Bacillota and Pseudomonadata, with 4 genera: Bacillus, Enterobacter, Mycodiria, and Bacillus. And CSC soil sample from Cibinong, West Java consists of 2 phyla, Pseudomonadata and Bacillota, with 3 genera: Bacillus, Parabrucodiria, and Brucodiria. The results show that the RM soil sample consists of various genera and indicated that RM soil sample has a high diversity of cellulitic bacteria. The phylogenetic tree of cellulolytic degrading bacteria separated into three trees. First, the phylogenetic tree phylum Pseudomonadota consists of 6 genera with 9 strains. 2 strains were identified as Glucoderia sp, 3 strains as Enterobacter sp, 1 strain as Phosphingobbium sp, and 3 strains were identified into species level: Parabrucoderia caribensis, Mitsoria ctenophorans, and Lysobacter enzymogens. All strains belong to the gram-negative bacteria. In this study, Glucoderia species were obtained from implanted grassland in Central Kalimantan and West Java. This genus was reported to be main cellulitic bacteria with an important role in cellulose degrading in Japan and in China. Parabrucoderia caribensis also reported as the cellulitic bacteria. This species was first identified in 1999 as an exopolysaccharide producing bacteria and obtained from fatty soil under Digitaria decumbens grassland vegetation in France West Indies. Enterobacter sp inhabit a wide range of environment from soil, waste, biomass, and animal digestion tracts, and some of them have the ability to degrade cellulose. Phosphingobbium, Mitsoria, and Lysobacter are gram-negative bacteria that report have the ability to degrade cellulose. The phylogenetic tree of phylum Bacteroidetes show that 2 isolates belong to this phylum and closely related to Pedobacter sp RM40 and Mycobacterium cI RM157. Genus Pedobacter is reported to have cellulitic activity with high capacity of cellulose hydrolysis and cellulitic activity. A study reports that Mycobacterium sp DSB1 showed cellulitic activity and potential to be cellulose producer in the aerobic digest of a convert biomass into biogas. In this present study, Mycobacterium cI report to produce cellulose activity. However, the ability of this species not previously reported in the literature. Hence, this study is the first report of the cellulitic activity of Mycobacterium cI. Phylum Bacillota consists of 7 isolates and is identified at the genus level and 1 isolate at species level. 5 strains were closely related to Bacillus sp, while 2 strains were closely related to Paenibacillus sp and Paenibacillus clibensis. Genus Bacillus was first reported with cellulitic activity in 1984. This finding revealed that various genera from phylum of Pseudomonadota, Bacillota, Bacteridota, and Actinomycetota have the capability to produce cellulose. Further study on the quantitative analysis of the cellulose enzyme measurement is needed. Thank you.
Thank you very much, Miss Dini, for your interesting presentation. So we would like to directly proceed to our third distinguished presenter, Ms. Septiantina Diandriasari, with the title of her presentation is Identification of Stingless Bee and Heterotrigota itama colonies Splitting Techniques in South Kalimantan. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for the time that given to me. Allow me, Septiantina Diandriasari, to present the result of our research entitled Diversity of Stingless Bee and Heterotrigona itama colonies Splitting Techniques in South Kalimantan. This research, the researcher who was involved are myself, Septiantina Diandriasari, Safina Surya Hakim, Siswadi, Reni Setiawahyu Ningtyas, Beni Rahmanto, and Wawan Halwani. Okay, for the time, so we into introduction. The stingless bee is a type of honeybee that doesn't have a sting. Even though it's stingless, it doesn't mean it can't produce bee products. Stingless bees are a group of bees which have economic and health benefits. Stingless bees are capable of producing honey, propolis, and bee pollen. Many people in Indonesia have cultivated the stingless bee, and many people in South Kalimantan have doing the same thing. Many of these stingless bee cultivation business still use traditional methods by taking colonies from nature. This is thought to have caused a decline in the stingless bee population in the wild. One effort to overcome this problem is to find a colony splitting techniques that is suitable for the area of cultivation. Colony division, colony splitting is carried out so that the survival of the colony can be sustainable to support cultivation. But the other hand, the preservation of the colonies in nature is also running so that the population of stingless bee colonies in nature can be continued. The objective of this research is to observe the most suitable and feasible techniques in stingless bee colonies splitting, especially Heterotrigona itama in South Kalimantan. The methods, the time of the research is July until December 2019. With location, 3 locations in South Kalimantan province. The first location is forest area with special purposes, Rantau Tapin Regency. And the second is Gambah Luar village, South Hulu Sungai Regency. And the last is Layu village, Center Hulu Sungai Regency. The methods of sample collection for identification. It's taken from each natural colony for later identification. So the individual samples were taken by placing the plastic right on the door of hives, knocking the beehives, and then the bees will fly out from the hives. So we can catch out with plastic. Cotton ball with alcohol were put inside plastic and used to trap the bees, so that the trapped stingless bees immediately faints and dies. And then the stingless bee specimen was sent to the Zoology Laboratory Biological Research Center in Indonesian Institute of Science, LIPI. And then for the methods for hive material experiment, this study used 5 different types of artificial hives. The 5 materials are wood, bamboo, cement, clay pottery, and ceramic. Each artificial hive was repeated 3 times, so the total number of samples is 15. Then observe the development of the colonies that were placed in the artificial hive with each material. Observations were made for 5 months. And then the method for colony splitting, we used 2 techniques. The first is direct transfer colony splitting, and the second is indirect transfer colony splitting. For direct transfer colony splitting, is done directly by split the colony eggs or hives into 2 parts. The first part is the egg with the bee queen egg. So it's just the egg, the queen bee egg, and the second part is the egg or the brood with the bee queen. So we need to make a new queen with the 1 part. So we hope that in this technique we can splitting for 1 colony to be 2 colonies. And then for the indirect transfer colony splitting is by creating new line using a plastic hose. A hose is installed between the original colony on old hive and artificial hive, and then we hope that the stingless bee that want to enter the initial nest, and they will through the hose. So we hope we can stay in the new artificial hives like that. And then for foraging activity, the observation 2-3 times. There are in the morning and then afternoon and in the evening. What was observed, the number of individuals going in and out from the nest, then individual carrying pollen or not in the legs. Data were recorded and then processed using tabulation and descriptive. This is the result and discussion. So, diversity of stingless bee on South Kalimantan were identified 5 genus with 7 species. There were Geoniotrigona thoracica, Tetragonula levicev, Tetragonula minangkabau, Tetragonula fuscobelteata, Lepidotrigona terminata, Lopotrigona carnifrons, and the last is Heterotrigona itama. Tetragonula is the most abundant genus in Borneo, and this genus is the smallest stingless bee. The body is only 3 millimeters. Even though they are small in size, these bees are cultivated and produce promising beekeeping products. And then next, this is the result of beehive material experiment. This is we use 5 hive material, wood like this, and then this is from bamboo. This is the artificial hive from cement. This is from clay pottery, and the last is from ceramics. The result of this research related to be the hive material experiment is the wood have the greatest change and the success rate in the highest compared to other. These results were obtained by calculating the percentage of successful colonies surviving in the artificial hives from the first month until 5th month. So this is the wood. The stingless bee can be survived until the 3rd month, and then the 4th month is reducing. Maybe the temperature too hot, or maybe the rainy season, something like that. And then the next colony splitting test. The result of the splitting test can be seen that the direct transfer method has a fairly high success rate until 5th month. Meanwhile, the indirect transfer method has quite good with good initial symptom in the first time. So we can see this method, we can see that 100% is still survive in the 4th month, but the 5th month the colony was gone, died, or abandoned the hive. So we can say that the direct transfer is a good one. Is better than indirect transfer. And then the result of foraging activity can be seen that every material has a different activities. For wooden, the highest in the morning and decrease in afternoon. And then cement, this is high when it's afternoon, and then the decrease in the evening. Clay pottery is high in the afternoon, and then decrease in the evening. Bamboo, the highest activity in the morning and decrease in the evening. And then ceramic, the highest activity in the afternoon and then decrease in the evening. So this is the activity in out of stingless bee. It's affected by temperature in each hive. This is accordance with the statement from Souza Jr. They said that body temperature of the stingless bee is influenced by the temperature of the surrounding environment too. Okay, this is the conclusion. There were 7 species of stimulus bee that has been identified. There are Genio Trigona thoracica, Tetragonula leviceps, Tetragonula minangkabau, Tetragonula fuscobalteata, Lepidotrigona terminata, Lepotrigona carnivores, and Heterotrigona trictama. And then the colony splitting method by direct transfer performed best result compared to others. And beehive made from wood show the most stable percentage of colonies compared to beehive made of clay, bamboo, ceramics, and cement.
Okay. Thank you very much for your interesting presentation. So we finally proceed to our last distinguished presenters. Will be Miss Aida Wulansari. She will present her research with the title Growth Comparison of In Vitro Culture Sato Imotaro Colocasia esculenta variant antiquorum in solid and liquid media with increase in vitamin B level. Miss Aida, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, thank you for giving me the time to present my topic. My name is Aida Wulansari. I am from Research Center for Genetic Engineering, National Research and Innovation Agency, or BRIN. Today I'm going to share my study about growth comparisons of in vitro culture Taro 1 Imo or Colocasia esculenta var antiquorum in solid and liquid media with increase in vitamin B level. Taro belong to the monocotyl family of Araceae. 2 popular cultivated varieties of taro are Colocasia esculenta var esculenta, which has large cylindrical main tubers and classified as dozen type, and the second one is Colocasia esculenta var antiquorum, which has small globular main tubers and classified as Edo type. This Edo type also well known as Japanese taro or Taro 1 Imo. 1 Imo tubers is a source of high calcium and high fiber with low carbohydrate and zero sugar content, and well consumed as a food diet and is very good to help lower down the blood pressure, the blood cholesterol. Taro 1 Imo also has a low glycemic index, so it is safe to consume by diabetic patients. Taro 1 Imo is widely cultivated because of the high demand in domestic market as well as export market, especially to Japan. Japan requires 40,000 until 45,000 tons of tubers per year. The productions of high-quality taro seeds in a large quantity is needed to meet the market demand. Until now, taro seedlings are produced from the tillers. The number of tillers is not sufficient because of the limited number of the parent tubers of the Taro 1 Imo. On the other hand, the use of the tubers or parent tubers will also reduce the yields. As alternative techniques to overcome this problem is by plant propagation through tissue cultures. This technique is advantageous because it can produce seedling in large quantity in a relatively short period of time, free from pests and diseases, and it doesn't depend on the season as well as it requires less initial seed stocks. One of the most important factor in tissue culture is the culture media. There are 2 types of media, solid media and liquid media. For commercial purposes, automatization of in vitro technique is required to reduce the production cost. The use of liquid media is more possible for automatization compared to liquid media. There are 2 systems of liquid media, full immersion system and partial immersion systems. In some studies, the use of full immersion system often results in lack of oxygen supply that causes the shoot death or plantlet death. This doesn't occur in partial immersion system, which can provide more aerations or oxygen supply. One type of automatization partial immersions is temporary immersion system, or TIS. This is a semi-automatic bioreactor. In TIS, plant cell, tissue, or organ are submerged in a liquid media for some periods of time. In some species, the TIS could increase shoot proliferations as well as enhances physiological processes such as photosynthesis and respirations. So the purpose of this study was to compare the growth of Taro 1 Imo or Colocasia esculenta var antiquorum shoot cultures in solid media, liquid media in pulse, and liquid media in this bioreactor using pre-selected media composition from our previous research. For plant materials, we use in vitro shoot of Taro 1 Imo collections from the Cellular Engineering Research Group for Plant Metabolite Improvement Research Center for Genetic Engineering BRIN. We use completely randomized design with two factors. The first factor was type of culture media, solid, liquid in pulse, and liquid in TIS. And the second factor was media compositions, media A, B, and C. In A media, containing MS with 2 mg per liter BAP, 2 mg per liter adenine, and 1 mg per liter thiamine. In B, it consists of MS with normal concentrations of nicotinic acid and pyridoxines and 100 times higher concentrations of thiamine than the normal concentrations. And the C medium consists of MS with 100 times concentration higher than the normal concentrations of thiamine, nicotinic acid, and pyridoxines. The data was statistically analyzed using ANOVA and further tested by DMRT with alpha 5%. In solid culture, we solidified with 4 gram per liter of Vita gels and we replicated 3 times. Each bottle consists of 3 explants. In liquid in pulse, we use 100 millipulse containing 10 milli media with second at 100 rpm, and we replicated 3 times, and each pulse contained 3 explants. In liquid TIS, we fill the chamber with 200 milli media and soaking interval every 4 hours with 2 minutes of soaking time, and each chamber containing 10 explant, and we replicated 3 times. All culture we incubated in a culture room at 25 and 26 degrees Celsius with continuous photo periods. Growth parameter observed 4 weeks after cultures. At the 4-week-old plantlets, we acclimatize in the greenhouse with the media is a mixture of soil, cocopeat, and roasted husk. From the charts on the left side, we can see that the highest value of shoot number was shown in the solid culture type at media A and B, followed by liquid in TIS and liquid in pulse. From our previous research, media A was the best media for micropropagations of diploid taro. The additions of 2 mg per liter BAP in media A could increase shoot proliferations. In media B, there was no additions of BAP, but contained thiamine 100 times higher than normal concentrations. Number of shoots in media B was not significantly different from shoot number in media A. The increasement of vitamin B level concentrations until 100 folds from normal concentration at media C did not influence the number of the shoots. On the right side is the highest value of the petiole was shown in the liquid pulse type cultures at media A. All other media composition gave lower petioles. Increased petiole growth may be because of the interactions between liquid media, BAP, and thiamine 100 times higher than normal concentration of MS vitamin. Interaction between BAP and thiamine stimulate cell divisions and growth. On the right side chart, we can see that the highest value of leaf numbers was shown in the solid type in all media and in the liquid in this culture type in media B and C. The leaf growth had a similar pattern like the growth of shoot as previously explained. On the right side chart, the highest fresh weight was found in liquid in this culture type at media B. It looks like adenine and BFP did not give significant effects to fresh weight in all three types of cultures. This is the root percentage and number of roots. The highest percentage and number of roots was produced in the liquid false culture type as shown in these two charts. Solid media gave poor rooting except with increasing high vitamin B level as shown in media C compositions. The percentage of root growth reached 100% in liquid false type cultures with media A and B. On the other hand, at solid culture type, with the same media compositions, media A and B, show the lowest root percentage. This is the performance of shoot cultures. On the top right is the shoot cultures after 4 weeks in solid type cultures in the first row, and the second row is liquid in false, and the third row is the liquid in TIS. On the left side, on the top left side, is the 4-week-old plantlet before acclimatizations. On the bottom right is the acclimatized Taro 1 Imo plantlets after 2 weeks in the greenhouse. As conclusion, Taro 1 Imo shoots grew well in solid, liquid false, and liquid TIS in MS medium with increase in vitamin B level. Increasing vitamin B level in solid and liquid TIS was better for shoot growth and biomass of Taro 1 Imo. Rooting was better in liquid false culture type. However, this did not affect growth of plantlets in ex vitro. Solid culture type can be used for initial cultures, and then shoot can be transferred to bioreactor TIS for further better growth. And the plantlet growth in TIS had better survival rate compared to solid and liquid false media. This combination technique may be adapted for mass productions of taro cultures. I think that is all.
Okay, thank you very much for Miss Aida for the interesting presentation. So all of our distinguished presenter already finished their presentation, so we could proceed to the next session. That will be the Q&A session. So I know that the participant here may be very excited to ask the question to our presenters here. So there is 2 types of way you could convey your question. First, you could raise your hand by clicking the raise hand icon on the Zoom application, or you can type your question in the chat box. So we already have first question in the chat box, which is from, wait a minute, from Miss Carla. The question is directed to Miss Dini Hari. So the question is, the result is very interesting. How to isolate cellulitic degrading bacteria and what is the medium? So Miss Dini Hari, are you there? Hello, Miss Dini Hari. Your voice is not clear. Maybe the connection is not good. So, Miss Dini Hari, could you try to say something?
Oh, yeah. Can you hear me, Miss Tina?
Oh, yes. Now I can hear you. Please directly address the question.
Okay. Thank you, Miss Carla, for the question. I'm trying to answer it. For isolation the cellulitic degrading bacteria, I isolate this bacteria by using 4 selective media such as Yema, Picoskaya, Caceres, and Sucrose asb. So all isolate that I obtain I screen for the PGPR traits like hydroacetic acid production, phosphate solubilization, nitrogen fixation, and then cellulolytic activity. For cellulolytic activity, I use the MC media for screen their ability to degrading cellulose. And this study, this is my previous study and already published in other conference.
Okay.
Okay.
Thank you, Miss. Okay, thank you, Miss Dini Hari. Miss Carla, are you satisfied with the answer, or you want to ask other question?
Oh, thank you very much, Ms. Dini Hari. It's very interesting. Can I ask one more question? And for example, if we go to the field and we want to collect, and then do you have any suggestion how we pack or how we bring the sample? So maybe when we collect from the field, the characteristic, or it will be different when we try to observe it in the lab. Do you have any suggestion?
Okay, I'm trying to answer. So you asked about the soil sample in the field, right? And then I did in the laboratory.
Yeah, right.
For soil sample from the field, I don't know, because this is a part of research, big research. So I just got the cell sample in laboratory, and I did to isolate for PGPR to obtain plant growth promoting rhizobacterium, and I screen for all isolate I obtained. And then Ms. Carla, sorry, can you hear me?
Oh, yes, we still can hear you, Miss Dini Hari. Please continue.
Then for the isolate in laboratory, isolate in the laboratory, I'm sorry, I don't know what you mean, Ms. Carla.
Okay, that's enough, I think, because I already get how to bring the sample from the field, but you have said that the one that take the sample is not you, so that's fine. So I thank you very much. Then I wish you luck with your research, then, Ms. Dini Hari. Thank you.
Oh, yeah, thank you. Welcome.
Okay, thank you, Miss Carla, for your questions, and also thank you, Miss Dini, for your answering the question from our participants. So we already have second question in the chat box, which is from Miss Santiratna Komala. The question is addressed to Miss Septiantina. The question, there is two questions. The first question is, how old colony does it take to splitting colony the most recommended? And the second question is, the old colony after splitting, can it still growing? Okay, Miss Septiantina, are you there?
Yes, I'm here. Thank you, Miss Pina. I will try to answer the question from Miss Santi. How old colony does it take to splitting colony the most recommended? Actually, the colony we take from nature or wild colony, we can directly to split that colony, and then after that we need to make the splitting colony did not noise with anything. So we need 3 months to make the colony can be held again, and then we need 6 months to harvest the honey and then the propolis or the bee pollen, something like that. But after we harvested the colony, we have to make the colony silent, or we do not disturb the colony after 1 month. After that, we can open that nest, artificial hives, and then we can see growth, the growing of the colony. Maybe like that, my answer. So we can directly split the colony if the colony from the nature. Okay,
thank you. Thank you, Miss
You're welcome.
Septiantina. Miss Santi Ratna Komala, is it enough?
Yes.
The answer?
Yes.
Oh.
Okay, thank you. You're
Okay,
welcome,
thank
Santi.
you. Yeah, thank you, Miss Santi Ratna Komala, for the question. We still have plenty of time in the Q&A session, so I hope that if there is anyone here that want to ask the question to our distinguished presenter here, you could still convey your question through the chatting box, or you could also directly ask by using the raise hand icon in the Zoom application. So while we waiting for the question from the participants, I would like to ask the question to Miss Aida Wulansari. So in the presentation, you stated that the TIS technique could decrease the cost. So my question is, what percentage of the reduce of cost that you could get if you change into the TIS technique, and what is other advantages by using TIS techniques? Miss Aida?
Yes, thank you. I'm sorry I turned off the camera because of the signal.
Okay.
Thank you, Miss Vina, for the questions. For the effective of the TIS or temporary immersion system is because it is automatization. It is different from if we use the solid media. If we use solid media, we have to subcultures every month. So the shoot or the plantlet will proliferate. If we use TIS or temporary immersion system bioreactor, we don't have to subcultures the media because of the automatizations of the bioreactor. And the other advantage for using TIS is it is more economic because of because of the use of the liquid media and for the automatizations. I think that is.
Okay, so in conclusion, we could shorten the time after using the new TIS technique, right? Thank you very much,
Yes.
Miss Aida Wulansari,
Yes,
for
thank you, Miss Vina.
your answer. And then finally, we can get the new question in the chat box. Thank you very much, Miss Safina. So the question is directed to our first presenter, which is Miss Carla Frieda Panto. So the question is, in the conclusion, it is mentioned that vegetation was dominated by dipterocarp. Regarding this conclusion, is the result applied for all plant stages or only for particular stage only. So, Miss Carla, please address the question.
Thank you, Ms. Safina, for the question. Yeah, in our research, because it's only a short time research, so the research is only in the stage of tree, because for the seedling and sampling, it takes more time, and then because the area is also not very easy, and then we focus on the tree. Okay. Thank you.
Thank you very much, Miss Carla. Is it enough, Miss Safina, the answer from Miss Carla? Okay, I hope Miss Safina is already satisfied with the answer. Is it interesting, because I also have question for Miss Carla, that is how to prevent from the scarcity of the vegetation, Miss Carla. Do you have any input or opinion about that?
Yeah, thank you. Actually, that's really a problem, how to conserve this plant. Yeah, the area in this national park is very large, and then the staff is only in small number. So that's a problem really, because there is lodging and also mining, and also some forest fire, and then maybe it's a small things that we can do. We can do like ex situ conservation. But I think in a university in Pontianak, in Untan, they already start this ex situ conservation. But I think I don't know whether in Kebun Raya Bogor there is also this species. I hope there is also. And then if there is representative in the small herbarium, and then maybe we can use the technology like tissue culture or genome editing, so they will not be extinct, I think.
Nice. Thank you very much, Miss Carla, for your input. I hope we can preserve more of the vegetation in Indonesia. And then we have other question in the chat box, which is also from Miss Safina. The question is addressed to our third presenter. So the question is, according to this research and your knowledge, is the splitting technique could be applied in the bee farming? Is this method have already practiced by farmer? So, Miss Septiantina Diahryendrasari.
Okay, thank you so much for the question. Of course, this splitting technique can be applied in bee farming. So, actually, for direct transfer splitting colony, it used by bee farmer to split, and it's still using until now. So, I think that's the easy method to make one colony to be two colonies. So we just take a half of brood, and then if we can find the queen bee egg, we can take it too. If not, we can just take a half brood and a queen bee in the new artificial hives. And then but sometimes maybe from if for indirect we need to patient to waiting the colony can move to the new artificial hives because it takes 3 until 6 months to make the colony move to the new artificial hives, some things like that. Thank you so much.
Okay, thank you very much, Miss Septiandina. I hope your question could satisfy Miss Safina, or if Miss Safina not yet satisfied with the answer, you also can ask next question in the chat box or could also raise hand. So we still have time actually for the Q&A session. So while waiting the question for all of the participant here, I would like to ask actually Miss Septiantina that is there any specific reason for the selection of the material for Hive B? You selected 5 material, right? So is there any reason why you selected this material?
Okay. Beehive actually, Miss Vina.
Oh, sorry, beehive.
Yeah, beehive. Okay. I take that material because I actually looking for survey from the other bee farmer like that. They usually use artificial hives from wood and then from bamboo, and then actually the stingless bee makes a nest at the what we can say, the door from the farmer, something like that. So I want to know how the strength, how the strong the colony, and then the preference the colony to make a new hive, something like that. So I want to see that. So maybe that's only my result from observation in the field. So the farmer used the wood, used the bamboo, and then the cement, and sometimes make a nest at the water clay, something like that. So okay, I think I use that material to my research like that, Ms. Vina.
Okay, interesting. Thank you very much, Miss
You're
Septiantina.
welcome.
I think we have no other question in this session. And also, unfortunately, the time is done for this session. So I would like to say thank you for all of the participants here. And I would like also say thank you very much for our distinguished presenter for giving us and sharing the knowledge with us. And yes, this is the end of this session. So I hope you can gain something from this session. Thank you for joining this conference. So I would like to give back the time to the committee. Thank you very much.
Thank you.
Thank you.
Thank you, Dr. Vina. So for the next session, all the participants can go to the main room.
Recording in progress.
Welcome back to the main room, ladies and gentlemen. Now, we would like to invite Ms. Novio Ayusril, Product Specialist, Protamix Nutrilab, to share the presentations. Ibu Novia, good afternoon. Thank you for joining with us. Can you hear my voice?
Hello.
Yes, time is yours.
Thank you, everyone. Give me one moment, please. Yes. Do I need to present in English or should I just in Bahasa?
It will be better in English, Ibu, because the participants not only coming from Indonesia. Thank you.
Okay. Thank you. Yeah. Good afternoon, everyone. My name is Novio. I'm a product specialist. Nutrilab Pratama is one of distributors bio life science products in Indonesia. So our office is in Kebayoran Lama, South Jakarta, and here you can see you not only can find us in Jakarta, but we also have office like branch office in Bandung, Semarang, Yogyakarta, Surabaya, and Bogor. So for BRIN especially, we have ourselves here. We will help you guys whatever you need from us. So you can see in the right side that we can support you guys with these products, but mainly for Nutrilab, you can reach to us for Gitco product, Invitrogen, and Thermofisher scientific products. This one especially for you guys who are doing biology or biomolecular research and also cell culture research. And also, not only that, we have several products which support your like general instruments such as centrifuge, cortex, etc. So we have also bio-based and also Hanil from Korea and Clever Scientific from UK. For the plastic consumables, we have products such as TPP, Tarfon, Nest, etc. So this is the short introduction from me for our company, and maybe I will move to the next slide about what is the product or what can we do or what can we help you with your research here. So we have several divisions in our company, mainly supporting this group of research. First one is molecular biology. You guys who use PCR reagent or like real-time PCR, and also our best product is qubit, come from Invitrogen products. And also for protein biology, such as antibody, ELISA, Western blot product, especially for this product line, you can reach out to me if you want to have more information about protein biology. And then we also support you guys with synthetic biology from Thermofisher scientific. So if you're doing expression protein and also cloning, you guys can reach out to us how to order, or you want to try like positive control for your real-time PCR, you can also reach out to us. And then cell biology, our main product, which is from Gitco. So we are the only one, like the only authorized distributor for Gitco product line in Indonesia. So if you are doing cell culture, you can also reach out to us. And then the last time is cell analysis product, which is microscope, fluorescent microscope, flow cytometry, and also high content screening. This one is the for protein analysis flow. As I mentioned before, that anyone who use protein like to do such analysis like Western blot, mass spectro, we have these many products that can support you guys. So from the extraction of the sample to the protein cleanup like dialysis, desalting, concentration, and also the downstream applications such as I mentioned is the stage Western blot workflow. We have here like Western workflow that maybe you guys also have heard before, that usually you takes 2 days to do Western blot, right? We have product line that can use that you guys can use only in 4 hours. You can get the result. So if you guys are interested, you can reach out to us for this application. And the last time is the analysis. Next is PCR workflow. As you can see here, we have we can support you the whole workflow from the extraction of DNA or RNA and then the quantitation. So here you guys may be familiar with nanodrop or spectro. So here we have a qubit fluorometer which use fluorometer detection. And also we have the master mix for both conventional and real-time PCR. And then of course the PCR instruments, and also the electrophoresis system here which use power snap. Next, as I mentioned, that we support you guys with the GIPO cell culture media. Maybe you are familiar with our GIPO product already. So you can see that from the isolation and then to the storage media, you can see here that we can support you guys with this. This one is our like high-end product, which is high content screening. Maybe you are familiar with this this term, right? That you can use this microscope, fluorescent microscope light plus microplate reader, and also that analysis data for your drug discovery. So you can see that this, for example, you have like a chemical compound that targeted RNA or other target in your cells, and then you want to try to check whether this drug is actually works for your cancer or for other diseases. So you can try with our high content screening cell intake CX7 LBR Pro. And then maybe for the next level, you can also image the this work with bioluminescence. This is, I think, my last two slides for our general equipment, such as I mentioned before, right? The freezer, centrifuge, like UV cabinets, and also all the stuff that you are familiar with in your lab. You can also, if you want to purchase your unit, you can reach out to us, or maybe some consumable or like accessories that you need, you also can reach out to us. I think this is my last presentation. So this one not only for bioscience products, we in Nutrilab also support you guys with bioproduction products. Maybe next time, now you are doing like the R&D step, right? And maybe in the future you want to produce your own like vaccine or your own drugs or protein recombinant. We can support you guys with these product lines from research development, bioreactor or fermenter. Also, we have this for biofactoring. Maybe you guys doing cell culture, which like adherent cells. You can also use our biofactory from next. And the last time is for purification of your protein in large scale. Usually you just only use like thin column or ACTA, right? You're familiar with those stuff. Here we have the resin, which can help you guys to use it in your purification system. Maybe that's all from me. This is I'm very grateful for this opportunity to present to you guys all our products in Nutrilab Pratama. So thank you so much. Yeah, see you guys.
Thank you very much, Ms. Novio, for comprehensive explanations. As what mentions, ladies and gentlemen, you can contact NutriLab directly for further informations of their services and also their products. So, distinguished guests, ladies and gentlemen, as the time is now 12:22, we will have a break for approximately an hour and 8 minutes, I guess. And please kindly back to the Zoom meeting at 1:30 p.m. Jakarta time. Once again, we will have a break for approximately an hour and 7 minutes from now on. So please kindly back to the Zoom meeting at 1:30 p.m. Jakarta time. Thank you and have a nice lunch.
Hello, Anna.
Hello, Uni.
How are you?
Hey, fine, fine. How are you?
Very
Good to
good.
see you.
Yeah.
Great, great.
Yeah. So what's your? Are you already okay now?
Yes,
I
I'm
mean,
fine. Yes.
Okay. Okay. So how long?
Shall I just
Yeah. Start your
share my screen?
Yes. Yes, please.
Because I'm trying to do that with two computers, so I have to see how this is working. Just a second.
Yeah.
Yes.
Stop sharing ya, Mas. Enggak, share screen. Are you familiar with Zoom, Arnold?
I'm sorry?
Are you familiar with Zoom?
No, we are normally using mostly Teams.
Teams. Oh, okay.
But I try to share my screen.
Yeah.
But I cannot. But I get the message. You cannot start screen sharing while the other participant is sharing.
Okay. So the organizer will stop this. Okay. Now it's already Okay, you can
now it
try.
should be possible.
Yeah, yeah.
Okay, let me just try. Yes.
Yeah, I already see your presentation.
Yes, but there's still some annoying thing in here.
No, it's
Let me
done.
check. There it is.
Yes.
Yeah, it's working. Perfect.
Okay.
Good. Now, I'm glad it's working, Uni. So this is the way we will do it.
Yeah, Arnold. So after the break, around 6 minutes before your talk, there will be a parallel session, and you can choose which room do you want to.
Yeah, I will check.
Yeah, yeah.
I want to go.
Yeah.
But just a second, I'm trying to get out of the screen sharing now.
Oh, gimana caranya kalau?
Because that's not working yet.
Ya udah. Gimana caranya kalau mau keluar dari share screen?
Here.
Yeah. Share. Share. So in
I'm
this
still sharing, eh? Or
No,
not?
no. You already stopped sharing your screen.
Okay, then it's fine.
Yeah.
All right. I guess it will work.
Okay, thank you very much, Arnold, for your participation.
No, I'm very happy to do that. Thank you for the invitation. It's nice.
Yeah. In this afternoon, there will be only you presenting in the plenary. Yeah, plenary speakers.
Yeah.
So you will have a lot of time for presentation and the discussion. Yeah.
Okay, fine. Yes. Great.
Okay, I'll not see you later.
See you later then. Okay.
Yeah.
Bye-bye.
Good afternoon, ladies and gentlemen. Welcome back to the NINF of ISIBio. The next agenda is continue the parallel sessions. Also, please kindly back again to the room, and you can choose the room based on your interests. Allow me to read the 5 rooms today. Room 1 with the theme of bioresources and biodiversity. Room 2, nutraceuticals and natural products. Room 3, biotechnology and bioinformatics. Room 4, environmental sustainability. And room 5, biorefineries and circular economy. Each room will be led by moderator, so please kindly click the feature of join breakout room. You can find out on the left corner of your Zoom. There will be a more and then join breakout room. 3 dots more and then join breakout.
So, good afternoon, dear distinguished participants and also organizing committee. Welcome back to our parallel session. I hope that you have a nice and delicious lunch, so then we can start our afternoon parallel session with more spirits and more joy. So in this session, we will have 4 presenters. They will talk about banana, coffee, and also bacterial research. And according to the list that I have, the first presenters will be Ms. Trihandayani, and she will present a topic about micropropagation of wild banana Musa acuminata cola using shoot tips and zygotic embryos. Before we start, let me introduce myself to you. I am Yustian Rofi Alfiansyah, a researcher from Research Center for Applied Microbiology, and currently I'm living in Bremen for being postdoc, and I meet the organizing committee as well as distinguished participant and also presenter. We start our presentation. For the first presentation is Ms. Trihandayani, or maybe her co-researcher will present. So I give the floors to you. Please present your presentation.
Thank you for the opportunity and the time. My name is Trihandayani. For ISIBio 2022 seminar, I would like to presentation with the title Micropropagation of Wild Banana Musa acuminata cola using shoot tip and zygotic embryo. For introduction, as we know that Indonesia are highly genetic diversity of wild banana, especially Musa acuminata cola species that hypothesize one of the ancestors of cultivated banana beside Musa balbisiana. There have been 15 varieties of subspecies of Musa acuminata in Indonesia that identified by Nasution, namely Malakensis, Sumatrana, Zepina, Microcarpa, and so on. Meanwhile, Bangsii, one of the wild banana that grows in Sulawesi and Papua, was not included in Nasution list. The germplasm of wild banana is a source of genetic diversity that can be used for genetic studies or genetic improvement to produce new varieties. The conservation and the utilization of wild banana, such for screening, phenotyping, or genotyping, can be started by providing plant material that is propagated in vitro. So the objective of this study was to determine the efficiency of aseptic culture initiation and regeneration of sick wild banana accession derived from comb and from zygotic embryo explant. For plant material, the wild banana seedling, sucker, or crumb, and seed from 6 accession taken from the germplasm collection in Cibinong, Bogor. The accession that we use in this experiment are Malacensis, Pandamensis, Breviformis, Sumatrana, Tomendosa, and Zebrina. Explant that derive from comb we divide into 2 types of explant. First is apical shoot bud, and the second is axillary bud explant. And explant from zygotic embryo, we extracted from the seed that the banana embryo is characterized by yellowish white solid small sphere that located in the interior of the seed cavity next to the micropyle that surrounded by endosperm, which is characterized by white powder. For culture initiation from shoot tip that derive from comb, first we sterilize the explant, then we planted the explant in antibiotic medium that contains streptomycin, rifamycin, and cefotaxime about 1 until 2 weeks. The uncontaminated explant we transfer to shoot induction medium that contain 4 mg per liter methyl adenine and 2 mg per liter indole acetic acid. Then the shoot that grow from the explant we transfer to multiplication medium that contain 2 mg per liter methyl adenine. The explant that were planted in medium containing antibiotic maintained in the culture rack without direct lighting. The second is for embryo culture initiation. First, we sterilize the seed and carefully we extract the embryo from the seed. Then we planted the embryo in the medium that contain 0.5 mg per liter methyl adenine for about X week. Then we transfer to shoot multiplication medium that contain 2 mg per liter methyl adenine for about another weeks. For embryo culture, we maintain in the dark room with the temperature is about 25 degrees Celsius for about 6 until 6 weeks. When the embryo germinated, we transfer to the shoot multiplication medium in the culture with the lighting for 16 hours. This is the data from our experiment show that the explant from apical bud have higher regeneration and lower contamination than the explant from axillary bud. The regeneration is varied between genotype or accession that rank about 20 to 75% higher when we use with the contamination rate for about 25 to 78% higher when we use axillary bud. The axillary explant that came from outer layer had higher contamination because the explant have greater possibility of direct contact with the soil, so the infestation of fungi and bacteria is more significant in the outer layer than the inner layer or from the apical bud explant. This is data for embryo culture from total 706 zygotic embryo that plant in germination embryo. About 52% degenerating into plant into shoot, and about 3% degenerating into callus, and about 35% became necrosis or contamination. Accession of Bantamesis and Malacensis have higher rate of degeneration into shoot than the other
accession. Embryo germinating into shoot begin with the embryo enlargement after 2 weeks, followed by development of elongated structure, which was become coleoptile after 4 weeks, and the root grow after that. From the data, by comparison of the wild banana genotype response, so that the genotype have different response in developing new shoot based on the type of the eggplant, such on Malacensis or Bentamensis have higher regeneration when use embryo eggplant, while Breviformis and Tomentosa have better regeneration when use corm or shoot-tip eggplant. In wild banana that produce many seed, in vitro propagation of plant can be carried out using seedling corm or embryo culture. That eggplant derived from sucker or corm are generally big. The main problem are the high level of contamination due to the larger size of eggplant and the phenolic compound that cause browning and the death of the eggplant. For the seed embryo eggplant, the main problem is the small size of the embryo, and the size is between 0.5 to 2 millimeter. Therefore, the isolation is quite tricky and could damage the embryo, and the different subspecies of wild banana have different size of the embryo. And the use of eggplant derived from this seed is likely to have different genetic variation compared to the eggplant from shoot tip that propagation vegetatively. So the conclusion is the propagation of wild banana can be initiated using shoot tip from corm and zygotic embryo eggplant. Using eggplant from corm had a higher level of contamination and lower shoot growth than eggplant from embryo. Shoot apical containing corm derived from eggplant from the center of the corm had a better regeneration response than eggplant from axillary bud. The average percentage of eggplant successfully regenerating into new shoot reached 79% for apical shoot and 39% for eggplant with axillary bud, and 16 to 80% for embryo depend on the genotype. The success of initiation of aseptic culture in wild banana subspecies depend on the origin of the eggplant and the genotype or assessing were used. Thank you. This research was supported by grant from DIPA, ex-Indonesia Institute of LIPI 2020 to 2021, and research program DIPA Rumah Program at this Organization for Life Sciences and Environment 2022. Thank you.
Thank you very much, Ms. Trihandayani and collaborators who already presented their research. And we will go to the second presenter, Mr. Fajaruddin Ahmad. I see that he is already in the room. And for the next, the third and fourth presenter, I hope that you will be here or already here. And so for the participants have question about the topic that first presenters already presented, I would like to ask to write in the chat or later on raise your hand. So then we can estimate the time and also distribution for the question and answer. So we go for the second presenter, Mr. Fajaruddin Ahmad. Time is yours.
Welcome everyone, welcome to my presentations in ISIBio 2022. In this program, I'm going to give a presentation about ploidy investigation using flow cytometry of Indonesian banana that we collected from our exploration program during period 2009 to 2019. So this presentation is on behalf of myself, Fajaruddin Ahmad, Yah Martanti, Ahmad Zailani, Herlina, Wicaksono, and Yuyu Purba. So Indonesia is known as center of diversity of banana. So in this country, we can find hundreds of cultivated banana and also the wild banana or wild relative. So cultivated banana is seedless, so it's edible. In contrast, wild banana is remain fertile and seeded, so it's not for consuming. So ploidy diversity in banana is because of alloploiploidy and because of autopolyploidy. So the alloploiploidy is because of intra and interhybridizations between two species, Musa acuminata and Musa balbisiana. So Musa acuminata is AA genome and it's also known pisang hutan, and Musa balbisiana is BB genome and is also in general is in people is known as pisang klutuk or pisang batu. And from these two species, it result diploid AA or AB, triploid AAA, AAB and ABB, tetraploid AAAA, AAAB, and also ABBB. So it's in common. We know that the example of AA banana is pisang mas, pisang oli, pisang rejang. AAA banana is such as Ambon and Cavendish, and tetraploid is quite rare. Autopolyploidy is the result of chromosome doubling using chemical, usually called piscine on oryzalid, to obtain tetraploid banana, or it's also possible to obtain triploid banana. So this is an example of chromosome doubling from pisang rejang. So this is the diploid one. This is from Purba et al. In 2016 publications. Using oryzalin, the chromosome is doubled into tetraploid. Ploidy investigations in general can be done using chromosome counting, morphometric, and flow cytometry. Chromosome counting is the most common approach. So this is, I think, this is the most precise to estimate the chromosome number, because we count the chromosome in the cell from the root tip or other meristematic tissue. But this approach is laborious. We cannot perform hundreds samples in a day if we make it by one person. So the other one is morphometric. This is developed by Simon and Shepard in 1955. So the analysis is based on the morphological characters such as the drooping level of the leaves, the shape of the male bud, the flower, and a part of the plants. This approach is not precise, but is still useful when we work in the field. So this is, I think, the latest technology to estimate the ploidy is flow cytometry. This method is fast and high throughput, so we can analyze hundreds of samples in a day. The aim of this study is to examine and confirm the ploidy level of the banana cultivar and wild relative from our explorations during period 2009 to 2019. So during the period of the exploration, we travel from Aceh to Papua. We collect hundreds of cultivated banana and also wild relative. For the ploidy identifications, using flow cytometry, we use this machine. So this is Partec Sci-Flow Space flow cytometry. We involving 257 accessions that consists of 19 accessions of wild banana, 238 accessions of cultivar. And we use Musa acuminata subspecies malaxensis accession lipid as the diploid standard. So we set the diploid standard in channel 200. And if the histogram occur in channel 300, so it indicate for triploid banana, and if the histogram occur in channel 400, it indicate for tetraploid plants. So this is our results. 65% of our samples are triploid. So this is makes sense that most of the cultivated banana that's planted by farmer or people is triploid banana, because this banana is the most has the highest economic value and mostly planted not only in the field but also in backyard. We found 21 accession or tetraploid banana. Tetraploid banana is quite rare; it's not common, but we still we still found some. And because of most tetraploid banana is usually is developed from breeding program by crossing triploid with triploid banana or triploid with diploid banana. And we confirm that all wild bananas are diploid. So this is more details. So this is the diploid banana. Is an example of diploid banana is wild Musa acuminata, Musa balbisiana, Mas banana group, Sang oli. And triploid banana, this is most common, such as pisang ambon, kepok, tanduk, raja, siam, and banana. Tetraploid banana is quite rare, but we found some cultivated ambon mandar, kayu biasa, tingalun, seblot, punti kayu, and lebu. And in this study, we reported a new finding of tetraploid siam, kepok, and banana. And this tree banana is usually known as triploid banana. Triploid and tetraploid banana in general look alike. It's similar based on fruit size, shape, and also the male bud color and also shape. But there is a morphological character that can be distinguished between these two banana. Tetraploid banana has more drooping leaf character, so like this, compared to the triploid one. So this is the pisang shape. And for the kepok is also more drooping compared to the triploid plants. Before I close my presentations, I would like to emphasize that the ploidy knowledge is needed for breeding strategy to obtain optimal progenies or targeted progenies. So this is an example of a breeding program scheme that's been done by IITA in Africa. So they cross tetraploid accession 917K2. So this is the tetraploid accessions, and diploid accession 912H3. So this is the diploid accession. And from this hybridization, they obtain accession 7798S2, or known as Narita1. So this is the triploid banana, Narita1. So the morphological character is different. It has a bigger bunch, more hands, more fruits. So it means that more productivity that will give a benefit for farmer. Okay, I think I'm in the end of my presentation. So I would like to acknowledge the contributor from this study, Program Competitive LIPI, period 2009 to 2015, Program DIPA LIPI during period 2009 to 2019, all member of Plant Genetic Laboratory, member of Tissue Culture Laboratory, Research Center for Biology LIPI, local governments in Indonesia, Ministry of Agriculture, and all peoples that involved in this study that I cannot explain one by one. Thank you.
Okay, thank you very much, Dr. Fajaruddin Ahmad, for the nice presentation. So we move forward for the third presenters. I suppose that the presenter is already here. So the topic will be about current genetic diversity of coffee species on Gayo Highland in Aceh, Indonesia. The presentation will be given by Mr. Muhammad Iksan Sulaiman. However, the lead author, Ms. Rita Andini, will present on this parallel room, and I would solicit to turn on your camera so then the participants and the committee can recognize you. So, Ms. Rita Andini and teams, the time is yours.
Hello everybody. Our presentation at this session is about current genetic diversity of coffee species on Gayo Highland, Aceh, Indonesia. Our team consists of Rita Andini and Ahmad Zailani from Genetic Engineering Research Center BRIN, Heru Pranowidayat, Muhammad Iksan Sulaiman, and Murna Muzaifa from Syah Kuala University, and Rahman Jaya from Badan Pengkajian Teknologi Pertanian Aceh. The presentation will arrange as follows. Ladies and gentlemen, Indonesia belongs to the biggest world coffee producing countries. More than 80% of the coffee bean are exported to the world. For this reason, coffee is an important commodity for Indonesia. Coffee belongs to the Rubiaceae family, and coffee arabica is the only tetraploid species compared to other varieties. In the world, we know 3 commercial accepted variety of coffee. The Robusta, Arabica, and Liberica, and the Arabica grows well in the higher elevation, while the other in the lowland area. In the world, coffee is growing in the tropical countries along the equator, between the Cancer and Capricorn. Aceh, located here in the peak of the Sumatra, is one of the biggest coffee producer areas in Indonesia. The coffee was introduced by the Dutch during the colony era. Nowadays, Gayo Highland in Aceh is the biggest Arabica coffee producer in Indonesia. The world market Arabica coffee from this area is known as Lintong, Mandailing, or Gayo coffee. In the Gayo Highland, most of the Arabica coffee comes from the community plantation, with the average area of 2 to 3 hectares per household in the elevation from 800 to 1,700 meters above sea level. Around 85% women involved in the coffee producing, from harvesting, processing, post-harvest handling, and marketing. The biggest harvesting period is between October and January. From the fresh cherry of coffee fruits to the green bean, coffee needs to be handled through the process of the pulping, fermentation, washing, drying, sorting, and grading. There are 3 well-known of the coffee processing. First is the natural process, where coffee is directly dried after the pulping. Second is semi-wash process, where farmers in Gayo are practicing. And the last is full wash processing, that will produce the best quality of coffee, but the process itself needs time and quite amount of water, therefore high cost and not popular among the farmers. Why this research important? In the field of Gayo Highland, deforestation is quite intensive. Many forest area is converted to the coffee and horticulture plantation because of price attractiveness. Second, the temperature is rising, lead to intensive pests and disease, as well as the Arabica plantation is going to the upper land, so that the cultivation area of coffee Arabica is reducing. Despite of the above situation, the research about coffee is not intensive in Indonesia. There are less records regarding the coffee genetic diversity, while farmers recognize high diversity in morphological characteristics. In spite of Arabica and Robusta, many is thinking about crossing of the both variety, producing Arabusta. So the aim of our research is to measure the diversity of Arabica coffee in Gayo Highland from their morphological characteristic and genetics through DNA. The second is to distinguish the Arabusta type of coffee and validate their existence due to the relatively long integration process from the Dutch colony era, long time ago. We collected the leaves from different altitude of Gayo Highland during the July and September 2021 by using about 10 field enumerators. There are totally around 62 collections that are collected from BPTP field station in Benar Meriah, as well as from the community with different local names, as well as the exotic collection based on the observation of the enumerators. We collect also Robusta samples from lowland area in Lamno, Aceh, and North Sumatra. This is the procedure of the genetic analysis of the coffee leaves. We use 13 SSR primers, and the result from the genetic analysis, we found quite high diversity of gene among the Arabica coffee from Gayo Highland area with genetic distance from 0.39 to 1, and we are grouping to 5 clusters. So for the discussion, although the Arabica coffee is not a native from Gayo Highland area, it was introduced by the Dutch with only several amount of variety, but the genetic diversity is quite high. It seems that farmers in the area is actively participate in the selection and conservation of the cultivars despite of many dynamics and challenges in the area due to social conflict, deforestation, and climate change. Further sequencing analysis is required to confirm the existing of so-called Arabusta, which is very essential to trace the coffee seed tree origins. As conclusion, molecular markers is still strong tools to elucidate the genetic diversity, particularly in coffee. Further in situ and ex situ conservation efforts should be initiated from now on by the support of all stakeholders, despite rapid change of climate and deforestation. So, we end our presentation, and we would like to express our gratitude to Syah Kuala University for the funding as well as to the enumerators and farmers in Gayo. Thank you very much.
Okay, that is the third presentation. And now we will go for the last presentation for this session, which will be delivered by Miss Santi Ratnakomala. So she and her collaborators did a research on antimicrobial and enzymatic activities of actinobacteria isolated from rumen fluid, and she will present her research for us in this session. And then after her presentation, we will start with question and answer for the all presenter. So I suppose Miss Santi Ratnakomala is present already here, and
Yes.
then time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good day, ladies and gentlemen. It is my great pleasure to be here to share our research. Today, I will deliver presentation with the title Antimicrobial and Enzymatic Activities of Actinobacteria Isolated from Rumen Fluid. It is widely known that Actinobacteria are an important and interesting group of gram-positive bacteria. Actinobacteria are the most successful known antibiotic producer and also various enzyme producer. Many species of Actinobacteria have been isolated from various ecological niches and explored to obtain new species of Actinobacteria that are expected to produce new species of antibiotic or enzymes. Although they are one of the most typical representative of soil microbes, they actively colonize and affect the digestive system of animals, including ruminants. There is a significant lack of information of the existence and biology of rumen Actinobacteria. Therefore, this study aimed to isolate unknown Actinobacteria for ruminal fluid and characterized from the bioprospecting. The rumen is a fermentation chamber, the largest stomach compartment in ruminant, which is bacteria digest the cellulose in grasses and feeds, converting them to fatty acid and amino acid, which are the fundamental used by the ruminant metabolism. Microbial fermentation in the rumen is fundamental to ruminant production and is driven by diverse range of bacteria, protozoa and fungi that work together to digest plant material and contribute substantially to ruminant host metabolism. Actinobacteria in the rumen gut of ruminants have not been extensively studied. Rumen microorganisms have a diverse range of enzymatic activity, cellulase, amylase, lipase, and protease. So, rumen is a good source for isolation of antimicrobial and enzyme producer Actinobacteria. Method in this research project, the source of Actinobacteria were obtained from rumen fluid samples by collecting rumen fluid from the rumen chamber, the cattle with fistula. Ruminal fluid was collected 2 hours after feeding and kept in a warm container until the isolation process in the laboratory. 29 isolate Actinobacteria were isolated by serial dilution and spread in HV agar medium and incubated at 28 degrees for 14 days. After that, we carried out molecular identification based on 16S RNA gene similarity. Then we screen the ability to produce antimicrobial against the test bacteria, Staphylococcus aureus and E. coli. Next, we screen the ability of rumen actinobacteria to produce various exoenzymes such as protease, amylase, lipase, and cellulase. As the result, 29 actinobacteria isolates were obtained from rumen fluid samples. 17 isolates were identified based on its 16S RNA gene similarity, whereas 12 isolates were unidentified. Of the 17 isolates, all of them belong to the genus Streptomyces, which consists of 8 species. Table 1 is about list of all rumen actinobacteria strains isolated from rumen fluid. This is a phylogenetic tree of rumen actinobacteria. It can be seen that actinobacteria isolated from rumen fluid are potential candidates for new species from the genus Streptomyces. Table 2 shows the clear zone index or CZI value of the rumen actinobacteria isolates against the growth inhibition of Staphylococcus aureus and E. coli as test bacteria. We can see that the isolate room 26 has the largest CZI value against Staphylococcus aureus with a CZI value of 1.5. And the isolate room 6 has the largest CZI value against E. coli with a CZI value of 1.12. The above results show that 12 isolates or 46% of actinobacteria isolates can produce broad-spectrum antibiotics by inhibiting gram-positive Staphylococcus aureus and gram-negative bacteria E. coli. 4 isolates exhibited antibiotic activity against Staphylococcus aureus only, and 7 isolates did not have antibacterial activity. This indicates that the gastrointestinal ecosystem is an attractive source for obtaining isolates of actinobacteria with antibacterial activity. Next, for bioprospection of actinobacteria producing enzyme. Here we screen 4 kinds of enzyme: protease, amylase, lipase, and cellulase. Table 3 shows the clear zone index or CZI value of the growth of rumen actinobacteria in medium agar containing substrates. Schema for protease, starch for amylase, twin 80 for lipase, and carboxymethylcellulase for cellulase. We can see that room 7 has the largest CZI value for production of protease with the CZI value of 2.59. For room 14 has the largest CZI value for production of amylase with the CZI value of 2.9. Room 10 has the largest CZI value for production of lipase with the CZI value of 1.17, and room 13 has the largest CZI value for production of cellulase with the CZI value of 1.49. Room 14 and room 19 can produce the 4 types of enzyme tested. The above results show that rumen actinobacteria are able to produce enzymes that vary in activity. Protease range from 0.35 to 2.49. Amylase range from 0.64 to 2.90. Lipase range from 0.46 to 1.17, and cellulase range from 0.49 to 1.46 respectively. Two actinobacterial isolates, room 14 and room 19, were able to produce the 4 enzymes tested. Ruminant digestibility is affected by the number of ruminant microbes. Bacteria can break down carbohydrates and protein fibers and play an important role in the fermentation of rumen feed. Ruminant microbes inhabit and grow in ruminant stomach to support the fermentation of ruminant feed. In conclusion, is as follows. Actinobacteria can colonize the rumen ecosystem. Although they are one of the most difficult representatives of soil microbes, they actively colonize and affect the digestive system of animals. The culturable and unculturable actinobacteria may be involved in feed metabolism in the gastrointestinal tract of livestock. The results show that Streptomyces was the most commonly isolated genus in the rumen. It is necessary to develop new techniques and media for isolating actinobacteria from different environments to increase the variety of actinobacteria that can be isolated. Actinobacterial community at the specific habitat in the rumen food ecosystem are potential as a source for isolation the taxon of actinobacteria. That is all the presentation. Thank you very much for your attention. Thank you.
Thank you very much for the presentation. So we already listened presentation from the 4 presenters, and now it's time for question and answer. We have 20 minutes for question and answer. Start from now. And first, I would like to invite all the presenters to open the video, so then we can recognize you. In case any problem with network or signal, you can turn off your video, by the way. And we have some question, and I would encourage before the presenter answer the question, maybe you can introduce yourself with 2-3 sentences. So then this is a time for you to present yourself, and also for the next 20 minutes, if there is no question and answer, maybe we can also proceed with like discussion to follow up the research, because I found that this is the good chance to connect with other scientists to make a collaboration in the future with regard to research that we already listened in this session. So I see in the chat there is a question from Miss Elo Rifki, if I'm not mistaken. So she has a question for Miss Trihandayani and Mr. Fajaruddin Ahmad. So let me read, or maybe Miss Elo, you want to ask directly, or if not, if yes, please open your videos and then I can see, or raise hand. Yeah, so then please ask directly.
Thank you, Mr. Christian, for the opportunity. Actually, I'm interested into early presenters, right? And Mr. Fajaruddin Ahmad already answered my question, so the question is left for Mrs. Trihandayani. I also worked in tissue culture before, and I'm interested in the work of Mrs. Trihandayani. First is what is the sterilization method you use for food explant? You say it, you use 2 explants there. And the second is when you use zygotic embryo, you need to extract the embryo from the seed, right? And I want to know that what is the number of embryo in each seed, and because you said it is tricky to extract the embryo from the seed, I want to know how you do it. Thank you. That's all, Mr. Yustian. Thank you.
Please, Ms. Trihandayani.
Hello. Thank you for the question. First, what the question is what the sterilization method used for the explant, right? We use ethanol 17% and 90% ethanol for sterilization of the both explant seed and the explant for banana sucker from the plant from the field. We just use that sorry, can I answer in bahasa? It's maybe more clearly.
Yes, please, no problem. I will translate later.
Yes, please.
Oke. Ya, saya sterilisasinya menggunakan kedua explant saya menggunakan alkohol 96% dan 70%. Dan biasanya tetapi untuk yang explant yang dari lapang saya biasanya merendam dulu explant di dalam larutan fungisida sama bakterisida sebelum dibawa ke laminar. Setelah itu sebelum pas kita potong kecil-kecil explant terutama yang dari tunas anakan yang dari lapang, kita menggunakan kita semprot dengan alkohol waktu pemotongannya. Jadi itu mengurangi kontaminasi bakterinya seperti itu. Jadi kalau untuk bijinya biasanya, sedangkan untuk biji biasanya hanya setelah biji diekstrak dari buahnya, kita bersihkan dengan air, setelah itu direndam saja dengan alkohol 70% dan 80%. Seperti itu. And the second question is how many embryo contained in its banana seed? The embryo is just one embryo in banana, just one embryo in one seed of banana. So how we can extract that won't get damaged the embryo? We can use a stereo microscope. So we can clearly see the embryo from the seed. So we can not cut the embryo, and usually we use scalpel with the sharp tip blade. So we can cut in the embryo. So, but not the embryo can not the damaged embryo. Like just maybe like that I can explain. Thank you.
So maybe Ms. Trihandayani, you can write your email in the chat, and then if there is a question, participant can reach you by email. And for the first answer, if I am allowed to try to translate in English. So for sterilization, there is a subsequent sterilization method using ethanol 70 and 90 before we go to or before we put or inoculate the explant to the medium. So for further detail, I would encourage to contact directly Ms. Trihandayani. So then if there is a question, there will be a detailed explanation either in English or in Bahasa. That will be convenient for the participant. And is there any more question for the presenters? So while waiting for the others, maybe I can raise question for Ms. Rita Andini and also for Ms. Santi Ratnakumala. So for Ms. Andini, my question is, could you elaborate the method to find the molecular markers when you analyze the diversity of coffee. And my second question, would the Arabusta coffee, if I'm not mistaken, the mix of Arabica and Robusta, that will be a nice and very delicious coffee. That's why I want to ask about Arabusta coffee, which the characteristic remains steady after several harvests, or there is a decrease of the quality. Maybe you can answer first, Ms. Rita, before I go to the next presenter.
Thank you. Thank you, Bapak Yustian, for the encouraging question. The first I would like to answer, I'm sorry, I caught a cold, so maybe my voice is a little bit, okay. The first question, we referred the work from Dimase et al. 2018, who had tried to distinguish the species diversity between the Arabica and Robusta, but in Ethiopia. We know that the birthplace of coffee in the world is in Ethiopia and still remains the same. But unfortunately, as I told you, even the coffee has been introduced to Aceh between the 16th and the 17th century. This is the first work where we are trying to distinguish the genetic diversity of Aceh coffee after more than 200 years. So, of course, this is a pioneer work, and there should be many improvement and enhancement. But we are delighted that at the end, the molecular tools were able to distinguish the genetic diversity existing in the Gayo Highlands. And regarding the second questions from Mr. Yustian, this is also actually my motivation, or our motivation, how to get the Arabusta, Mas. The problem when you go to the highlands, on Gayo Highlands, every farmer always say that they always plant Arabica coffee. Arabica is the high quality, the top sensory, the best one. Because you are a seller, so you always tell the people the good things, right? Because they want others to buy your product. So, but we as researchers hypothesize they are not Arabusta. Our hypothesis, maybe 70% have been already Arabusta, like you said. They have been already an integration between the Arabica and Robusta, because Arabica, as like people, is an elite person, but they are very soft, very weak. They are very easy to get diseases. So how can you imagine that Arabica, the pure Arabica from 16th century in Aceh, and they still remain as a pure Arabica? I think they have been already bred or already crossed with other person. Maybe itu Pak Yustian. Thank you very much.
Okay, actually, I still have a question, but I want to respect. I will follow later on. Maybe if you could write your email, that would be great. Okay. I would give a chance to Miss Wening, which write a questions to Miss Trihandayani. So, would you want to ask directly, Ms. Wening, or?
Okay, thank you for the question. For the seed embryo explant, we not use some tool aid. We just use if too difficult to see, we usually use stereo microscope, or maybe some seed is the embryo is clearly to see, so we just use a scalpel with the blood strap tip. We just cut the seed and we can see the embryo. And usually we see the embryo is under the microfile of the seed. So we just take the part of the seed from the under the microfile of the seed, like that. And which type of explant we suggest to use for wild banana is the second question. Usually it's depend on the purpose of the research. We usually want to use want to know maybe for genetic mapping or genetic, maybe example for genetic mapping we can use explant for seedling for embryo zygotic, or maybe if we want to know about the resistant or some pest or disease, we can use explant for the banana sucker or shoot tip, like that. Thank you.
Okay, thank you for the answer. Do you want to follow up this evening? Maybe by chat while waiting for your follow-up question. I have a question to Mr. Ahmad and Mr. Ratna, so then we will be equal for its participant. For Mr. Fajaruddin Ahmad, my question is, do you analyze your samples in pool or you analyze it individually? And how would you eliminate or reduce the noise of the flow cytometry method that you use? And after that, maybe directly to Ms. Ratna Komala, my question is, for the bacteria that you isolate, is it in anaerobe condition or in aerobe condition? So I would suggest Ms. Ratna first, and then we try to give a chance to ladies first, and then after that, Mr. Fajaruddin Ahmad can answer. I hope that you don't mind for this.
Okay,
so,
thank
Ms. Rat.
Okay, thank you very much. Firstly, I ask permission not to open the camera due to poor signal here.
Yes.
Regarding the question, actinobacteria isolated is we incubate in aerobe condition, not in an anaerobe condition. Yes.
Okay, in aerobe condition. Okay,
yes.
Thank you very much. Yeah.
Okay.
So now, Mr. Fajaruddin Ahmad, please.
Yes, thank you, Mr. Justitian. So yes, we did the flow cytometers one by one. It's not the pool, because I think that is the most ideal, because we would like to identify whether there is any variations among a cultivar. As I described in the paper, I think if you see the full paper later on, it can be much clearer, such as pisang kepok that we known as triploid banana. And in this study, we found that some accession of pisang kepok are tetraploid. I think this is a nice and a good result with information that there is a genetic diversity among pisang kepok itself, and also among Kapoxium we also found a tetraploid that is also known triploid banana, and also for Barangan and one cultivar I don't remember by heart, but you can check it in the full paper later on. I think it's smart. Okay, and then to reduce the noise during the flow cytometer, I think it's one of the trickiest things that we suffers in the beginning of our study using the flow cytometer. Yes, if we follow the protocol, we can add such as antioxidant agents, PVP or beta-mercaptol, or we can increase the concentration of PVP and beta-mercaptol. And based on my experience, we incubate the sample no longer than 5 minutes.
the protocol mentioned to incubate the sample for about 20 to 30 minutes, and I also talked with the technician from the flow cytometer company. They also suggest for 20 to 30 minutes. But if we, in my case, if we incubate the sample above the 30 minutes, the noise will be higher. Probably it's because of the reaction of the buffer or staining solution is much more intense with the other compound. And also the ploidy, the banana itself accession is also affects a lot to the flow cytometer. So flow cytometer, I menyarankan, yes,
suggest.
Yes, I suggest. Sorry, I suggest to use as young as part as possible tissue of the plants. So in banana, we use the cigar leaf that still remain inside of the stem. So we cut the stem, we collect the inside, or the very young flower from the male buds. So we cut into the male flower, the flower. We took the youngest part, and also part that that we think that less phenolic compound, such as the tip, the root tips is also good, but root tip sampling is not that convenient because we should dig the soil and etc. I think that is for my. Thank you.
Okay, thank you very much. So before we continue, I want to ask for the organizing committee for Pak Imam Surya, do we have additional time, or we stick to 39 for the maybe if I ask for 5 minutes additional time, would it be possible?
Yes, it's your. Actually, also I have a question, Mr. Faisal.
Okay. Yes. So please.
So, okay. If I'm not mistaken, that from your presentation, you showed that all the wild banana are deployed. So I was wondering that is it possible if wild banana is not deployed, maybe triploid or tetraploid? I mean, based on your experiments, have you already found that any wild banana is tetraploid or triploid?
Yes, thank you for the question. It's quite interesting. There is no report of triploid or polyploid banana, wild banana that found in Indonesia, based on my experience. But there is an indication. So I know a banana expert from Thailand, Dr. Hugo Volket. He is preparing a manuscript about his exploration in Thailand, and he found an indication a triploid banana from wild relative. So this is quite interesting because the appearance of the plant is look like a diploid banana, but the all seeds are sterile. So from this, it raises a curiosity to check with and count the chromosome at times. When I met him in Thailand about 3 years ago, and he found that an indication of triploid from wild banana. I think it makes sense because during the banana evolution, crossing inter or species or subspecies is quite common in banana. So in Papua, there is a cross between Musa acuminata and Musa sisucarpa and also Musa textilis. And in one subspecies, the gamete is failed to separate. So the gamete is remain diploid, and when it was pollinated or to be pollinated by haploids or diploid, other diploid pollens, so the progenies could be triploid or tetraploid. But because the level of the chromosome rearrangement is remain low, because the chromosome is derived from true wild relative, the gamete production is still remain like the wild one. So the pollen is still fertile, unlike cultivated banana. So the fertile banana, as I mentioned in the presentation, is because of the unbalanced segregations or rearrangement, and because of this still remain in a good shape, I can say like that. So it can produce a good pollen or a good ovum, and when it pollinated by other pollens, it can produce a fertile or viable seeds. So if we can see, the morphological character is look like a wild banana, which is produce seeds, pollens, and etc. But if we check the chromosome, it can be triploid, but it's quite rare. But the indication is there.
Thank you, Mr. Fajar. That is really interesting.
Okay. Do we have additional time, maybe 2-3 minutes?
Okay, please.
For question? Yeah. Or it is almost over? Because if it is almost over, then I will give a recapitulation of our session for a take-home message. Is there any chance to give another question? Maybe. Okay. Yeah. So then maybe one question for Miss Santi, sorry, Miss Santi, and also one question for Miss Rita. Maybe you can short answer my question. So for Miss Santi, are the capability of actinobacteria to produce enzyme is steady or stable, meaning that if you culture several times, they will produce similar profile of enzyme. And then for Miss Rita, if I'm not mistaken, the samples that you analyze is leaves of the coffee. Is it correct, or you also analyze others? Maybe we start with Miss Rita, which is already ready, and then we could.
Only the leaves, Mr. Yustian.
Thank you very much. So we go now to Miss Santi before I close the session.
Okay, thank you very much for the question. This is still preliminary study in this bioprospection of rumen actinobacteria. Further research is still needed to determine its ability, that is stable or not. I still to study about it. That is question answer. Thank you.
Okay, thank you very much. So we exit about 6 minutes. So let me give a 1 minute or 2 minute recaps for our sessions, and then we can get a take-home message. So from our presenters, we learned that there are several techniques that we can adopt, we can develop together. So that's why I encourage the presenters to write down their emails, and then the interesting study, or if a participant wants to follow up this meeting with joint collaboration or joint research, they can contact the presenter immediately. So we see that the genetic elucidation, I would say, is important to improve the quality of banana and coffee, and then the later on for bacteria as well. So we know the species, and then I think with the open access and open facilities in BRIN, we can collaborate later on. And then considering expertise that we have from each group of research that we already listened, we can make a collaboration, I think, if we want to work on genetic related study with the species that we interested in. For example, if we want to study into deeper for banana, then we know the expert also for coffee, and as well as if we want to employ microorganisms like bacteria for our needs of research. So then I congratulate for the presenter for their successful study, and then I apologize if there is a mistake from my side as a moderator who chaired this session. And for now, I hand on the time to the organizing committee, to Mr. Imam Surya and Mr. Dilan Lowry. Thank you.
Thank you, Mr. Yustian. So for all the participants, now it's time for the coffee break before to the next plenary session. So for the plenary session, you can go to the main room. Thank you.
Recording in progress.
Would you like me to already start sharing my screen? Would you like me to start sharing my screen already?
Not yet, sir. Or you want to try the technical matters right now?
Yeah, but I think, I can just try for a second.
Yeah, you can try before we start again at 3.
Just a second.
Yes.
Yeah.
Let's see.
Yes, we can see your slide clearly.
Yeah, that's it. That's fine.
Yes.
Okay, I'll stop sharing now then.
It's
and
okay.
you can put it on
you.
again. And let me see. Stop sharing. Yes.
Okay.
Good.
Thank you, Dr. Arnold. Menit lagi. Ladies and gentlemen, so happy to welcome you back on the main room. So the next agenda is plenary sessions. Therefore, may I invite Dr. Hayati Iskandar as the moderator of the sessions. But before that, I would like to read short profile of her. Dr. Hayati Minarsi Iskandar is a researcher of Indonesian Oil Palm Research Institute who are interested in molecular biology, biotechnology, and biochemistry. After graduated from Bogor Agricultural University or IPB, she continued her master degree in plant molecular biology at Vrije Universiteit Brussels, Belgium, and obtained her PhD degree in biochemistry and molecular biology at the University of Queensland, Australia. Without further ado, please welcome Dr. Hayati Iskandar. Dr. Hayati Iskandar, time is yours to lead the sessions.
Okay. Thank you, Ibu Aulia, for my introduction. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon in Indonesian time, distinguished guests, all the speakers, and all participants. Welcome to the next plenary session of the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBio 9 2022. Please allow me, Hayati M. Iskandar from IOPRI Bogor, as a moderator to lead this session. This afternoon, initially, we will have two guest speakers, Dr. Arnold Bovi from the Netherlands and Professor Chin Kun Wang from Taiwan. Unfortunately, Professor Wang is not able to attend this session because he suddenly has to conduct a surgery in a hospital. So we will start the plenary session with the presentation of Dr. Arnold Bovi. Am I right to pronounce your name, Dr. Arnold?
Yes, you're right.
Okay,
Absolutely.
thank you. First, I would like to introduce you about Dr. Arnold Bovi. So his CV. Dr. Arnold Bovi, he's a researcher at Department of Plant Breeding, Wageningen University and Research. He's also a group leader at Breeding for Quality Research Group, also at Wageningen University. And his research interest is metabolomics, plant breeding, and genetics. So Dr. Arno will present an interesting topic about application of genetics and metabolomics in assisting plant breeding for quality. So you will have 20 minutes for presentation, and we will have a Q&A session after that. So Dr. Arno, the time is yours. Thank you.
Thank you very much. I will now share my screen. Okay, well, good afternoon, everybody. It's a real honor for me to be invited to give a talk at this conference. I really would like to thank Wayuni and the other organizers for inviting me here. And well, the title of my talk in today's session is Application of Genetics and Metabolomics in Assisting Plant Breeding for Quality. And so, as was already mentioned in the announcement, I lead a research group on breeding for quality within the plant breeding department of Wageningen University and Research. And in my talk today, I will talk about the combination of genetics and metabolomics in order to assist breeding for quality. So we are in the plant breeding department. And well, actually, I would like to start with the question, what breeders want? What do they want in their breeding? First of all, they would like always to find novel sources of their traits. And in particular, they are looking for genetic variation, and in particular, crossable genetic variation with which they can, let's say, improve their crop with traits of interest. Breeders also would like to have tools to select for their traits. And on the one hand, these are efficient phenotyping tools, but more and more important in breeding is to have genotyping tools so that breeders can already select for a trait of interest at seedling stage by using molecular markers. And important for this story is that the best possible molecular marker actually is the gene that's underlying the trait of interest. In addition to breeding for yield and disease resistance, there's a growing interest to also breed for quality traits, and that's the topic of my work. And if you look at these quality traits, you can basically look at or see a number of traits that you can visually monitor, such as size, shape, color, and shelf life of your crop. But there are also invisible traits of interest, like nutritional value, taste and aroma, and pungency. And especially in order to monitor these invisible traits, we need a good detection platform. And many of these quality traits are determined by small molecules that are present in our fruits and vegetables, small metabolites, and detecting these metabolites is one of the main aims of our research. Well, we do that using metabolomics, and metabolomics is a technology aimed to provide a broad overview of the metabolic composition of a given extract. And metabolomics, as we use it, is based on mass spectrometry, which basically stems from the idea that every molecule has its own specific mass and can be detected as such. Well, what are the advantages of metabolomics for breeding? First of all, metabolomics can give you the insight in the metabolic basis of often complex traits. It also allows you to define subtraits. And defining subtraits is a way to simplify genetics, as I will show you in a minute. And also metabolomics and knowledge of pathways helps you in identifying genes that may be involved in this process. And to illustrate that, I would like to give you an example of a complex trait, taste, a trait that we worked on a lot in particular in tomato. And when you want to study the genetics of taste, the first level you can do that is by measuring taste appreciation using consumer panels. But you can imagine that just giving an evaluation of liking is not giving you a lot of differentiation in the traits. So a second level at which you can look at taste is at the level of trained tasting panels. And these trained tasting panels, they are able to discriminate many more aspects of taste. For example, they can dissect taste into, for example, taste, aftertaste, mouthfeel, and aroma attributes. And under these attributes, there are different sub-traits like sweetness, sourness, umami, and so forth. This is already a new level of detail. But if we would look at the metabolites underlying these traits, we can see that under each of these attributes, there are multiple players that contribute, multiple metabolites that contribute to this particular trait. So in fact, this very complex trait, taste, can be divided in many different subtraits by looking at the metabolites. And looking at these metabolites, studying these metabolites, also gives you hints on the biochemical pathways that are important and the genes that may be involved in different steps of these pathways. So that's a big advantage of studying metabolomics. Well, in addition to metabolomics, today's breeder's toolbox has really expanded. And it has expanded, for example, by the availability of genome sequences. And if you look at a crop like pepper, more than 400 accessions have already been sequenced today. In tomato, it's even more than 700 at the moment. There are high throughput marker systems available to genotype large numbers of genotypes. Transcriptomic tools and databases are available, and of course the metabolomics platforms. In addition to these tools, also there are more and more interesting breeding materials available. For example, the presence of mapping populations that you can use to study the genetics of your trait. A specific type of mapping population that I'm going to talk about today is a GWAS panel, which basically is a diversity panel of accessions. Tilling populations are available nowadays, so mutagenized populations that you can use to identify mutations in specific genes of interest. And of course, transformation technology, which is available, very important to, for example, study genes by virus-induced gene silencing or make stable knockouts using CRISPR-Cas gene editing. Well, in my talk today, the high-throughput marker platforms, metabolomics, and the use of GWAS panels is going to play an important role in the example I would like to give. It may be good to mention, in my research, tomato and pepper are the major crops that we work on, and the talk today, as you can see from the pictures, will be about pepper. The topic of my talk today was studied in a big EU project called G2P Sol, Genotype to Phenotype Sol. It is a large EU project that just ended at the end of last year. And the aim of this big program was to link genetic resources, genomes and phenotypes of solanaceous crops, tomato, pepper, potato, and eggplant. And my involvement was mainly in the pepper part of this work. The aims of this project was to do an inventory and conservation of material stored in gene banks. And it was mainly done by high-throughput genotyping. I will come back to that in a minute. A description, assessment, and phenotyping of the collection, genetic analysis of this phenotypic variation, and there was a pre-breeding part in this program. Well, I already mentioned that our main involvement was in pepper, and as I already mentioned, also genetic diversity that was studied is the diversity stored in gene banks. And for each of the crops, more than 10,000 different accessions were genotyped using genotyping by sequencing or other next NGS-based platforms. So for capsicum, 10,000 different capsicum accessions were sequenced. Most of them were capsicum annuum accessions, but also a number of accessions from Bacatum, Chinense, and Frutescens. And let's say the genotyping and the insights we gained about the evolutionary history of pepper has been published in this nice paper of Pascale Tripodi in PNAS last year. Well, based on the genotypic analysis and the genotypic diversity that was found, a core collection was made consisting of 450 capsicum annuum accessions. And this core collection was studied further as part of the project. And because this core collection was derived from the genotypic data, it was expected to represent the genetic diversity within capsicum annuum. And this core collection was further analyzed by high-density genotyping and by phenotyping of 3 trials conducted in different countries in Europe. Well, here you just get a small overview of the phenotypic variation that was present in this collection. So there's a huge amount of variation. Now, what we tried to do in the past year was to get a better understanding of the metabolic diversity in this pepper core collection. And if we think about the metabolic composition of pepper fruit, actually pepper makes a lot of very interesting compounds. Compounds involved in color formation like carotenoids, chlorophyll, and anthocyanins. Compounds involved in providing pepper with taste and aroma like volatiles, sugars, acids, and capsaicinoids. And other nutrients, dietary nutrients like vitamins, capsaicinoids, and capsiates. So we were aiming to measure these compounds in this pepper core collection. Now the trial that we conducted looked as follows. The collection consists about 400 capsicum annuum accessions, 20 chinenses, frutescens, and bacatum collections. They were grown at different locations, and I will talk today about material grown in France and grown in Italy. And ripe fruits of these trials were harvested and subjected to different metabolomics analysis. So basically, LCMS to detect semipolar secondary metabolites, and GCMS to detect volatile metabolites. Now, how many compounds can we detect using these platforms? Now, if we look at the GCMS platform that detects volatiles, we detected 128 different volatiles in the INRA trial in France and 133 metabolites in the trial in Italy. And most of the compounds that we detect, we can actually identify. And for the GCMS, we only had, let's say, 10 unknowns in total. With LCMS, it's a completely different story. In LCMS, we were able to detect, let's say, hundreds of compounds, but the majority of these compounds actually are unknown, and identification of compounds using LCMS is still a big challenge in metabolomics research. And the groups that we could detect consisted of capsaicinoids, flavonoids and other phenolics, and capsyanosides, which are diterpenoid compounds. Now, if we look at the distribution of these metabolites over the different accessions, you can visualize that using a principal components analysis. And in this principal components analysis you see here, actually all the dots represent the different accessions in our trial, and with the arrows, the main drivers of the variation are shown. And you can see a clear division in the first axis of the pungent genotypes on the right side, and the non-pungent capsicum onion peppers on the left side. And if we look at different, let's say, volatile compounds and taste-related compounds, we can see that on the right corner here is the place where most of the wild accessions end up, and they are, let's say, the richest in volatile esters, which usually give a fruity aroma to your fruit. So this gives you some insight in the metabolic variation, in this case for volatiles in this material. Now, there are hundreds of compounds that we can detect, but which are the ones to focus on? Which are the ones that are important, for example, for taste? Well, one way to do that is to see if you can find correlations between the presence of certain compounds and a certain taste attribute. And using a few colleagues, we basically determined the aroma and the taste of all these peppers. And we could discriminate, for example, 3 different types of aroma. The beany green pepper aroma that you find mostly in blocky peppers, a more grassy green fleshy cut grass aroma, and a sweet fruity aroma. And when we look at the correlations of these taste attributes with different metabolites, we can clearly see that methoxypyrazine is the compound that really is responsible for this beany green typical blocky pepper aroma. If we look at the grassy green type of aroma, it's mostly the lipid-derived volatiles that are highly correlated with this type aroma, and that is very well in accordance with also taste in tomato, where these compounds also play a very important role. And if you look at the sweet fruity aroma, it's especially a number of volatile esters that are highly correlated with sweet fruity aroma. So in this way, we can basically select a number of potential target compounds that we could use in breeding for taste. And for example, if you would like to breed for a pepper with a more sweet and fruity aroma, we would aim at increasing the amount of esters and decrease the amount of these methoxypyrazine or oxylipin-derived volatiles. So these could be potential breeding targets for taste in pepper. Well, we also tried to see if we could link the metabolic variation that we see to genetic variation. And to do that, we used a GWAS study, and GWAS stands for Genome-Wide Association Study. So basically what we try to do using molecular markers is to find associations between specific molecular markers and specific compounds. So the data we used for this genome-wide association study were these volatile aroma compounds and the secondary non-volatile metabolites, around 200 metabolites in total. We used the data from the 2 trials, from the 2 different locations, and we used SNP data from genotyping by sequencing, and we used in total 10,000 SNPs in this analysis. Now, we used also 2 different, let's say, standard approaches that are used in GWAS, mixed linear models and a blink approach. Now, in the GWAS, we could find a number of common QTLs that were found in both location trials. In total, we found QTLs for 105 metabolites at 524 different loci. And the first one I'd like to show you here is the GWAS analysis. GWAS results often are visualized in this kind of what we call Manhattan plots that here show you the distribution of the 12 chromosomes of pepper, and here the association of your compound with specific loci on the different chromosomes. And you can see here a very nice association at the end of chromosome 2 of capsaicin, the compound responsible for pungent taste in pepper, and this location on chromosome 2. And it's known that this location is the famous PUN1 gene, which is the gene that's the ester-forming enzyme responsible for the last step in the biosynthesis of capsaicin. And this very nicely matches with earlier results in which gene bank annotations for pungency also showed up a very nice QTL at exactly the same spot. Another observation that we did, and relevant for this story, is the fact that biosynthetically similar compounds, they correlate quantitatively with each other. And to show you that, we made this, let's say, this hierarchical cluster here, and each of the colors actually represents metabolites that are closely related to each other. And you can see that these metabolites form rather separate clusters. So it means that metabolites with the same origin or the same biosynthetic pathway are often correlated with each other, which would suggest that the regulation of these pathways may be regulated by hotspot QTLs. And the question is, do we see this kind of hotspot QTLs? And actually, when you look at this graph, here, this is actually a merger of all the QTL data that we found in our study. And we tried to visualize these data using bubble sizes. And the larger a bubble size, the more compounds are actually co-localized on the same QTL. So, for example, if you look at the bubble size here, at the position of the QTL for capsaicin, actually, we have here not only one different capsaicin, but a number of capsaicins that show the QTL at this particular position. And that is because this ester-forming enzyme, PUN1, does not only use one specific substrate, but has a broader specificity for different substrates, leading to different capsaicinoids. The same type of very nice bubble here, we see a QTL for different flavonoids. And we see this QTL at the end of chromosome 5. And our most associated marker was landing exactly in the MIP12-like transcription factor. And MIP12-like transcription factors are actually known to regulate the flavonoid pathway. And we cloned the MIP12-like gene from tomato a number of years ago and showed that these transcription factors indeed are regulating the flavonoid pathway. Well, actually in pepper, we find a very nice candidate gene called MIP12-like, which seems to regulate also the flavonoid pathway in pepper. The MIP12 gene in tomato is actually located on chromosome 1, and this ortholog is located on chromosome 5 of pepper. So we found quite a number of nice and interesting QTLs. But one of the main challenges of GWAS studies is to validate these QTLs, to make sure that they are really showing you the right lead and not false positive, for example. And one of the QTLs we validated was actually this MIP12-like QTL I was just talking about. And it was very nice to validate this QTL because we started that work already quite some years ago. And it was actually the work of Wayuni, and here you see a picture of Wayuni 10 years ago when she was still working in Wageningen, and she identified 2 accessions We call them here for simplicity P12 and P24. P12 was a very high flavonoid accumulating accession, and P24 was a very high vitamin E accumulating accession, but low in flavonoids. And in collaboration with Ilan Paran in the Volcani Institute of Israel, we developed segregating populations based on the cross of these two parents. And when you make segregating populations in the F2, these nicely segregate, and every F2 individual basically is a genetically unique individual. So we did a QTL analysis of this F2 population, and actually in this population, we also find a very strong QTL at the same position on chromosome 5. This QTL, you can see here that if you have the P12 allele of this QTL, you get much higher flavonoid levels than when you have the P24 QTL allele. And also what we found was that a large number of flavonoid genes showed a much higher expression when the P12 allele is present compared to when the P24 allele is present. And this, of course, suggests that there is a transcription factor into play here. And actually that also led us, independently of the GWAS trial, to the identification of the MIP12-like gene. Now, we did a number of experiments to indeed validate and show that this MIP12 gene is indeed the gene doing the job. One of the things we did is we knocked out the MIP12-like gene in this parent 12, the flavonoid accumulating parent, using virus-induced gene silencing. And you can see if you have the flavonoid level in the control, here plants that in which we used a GUS construct to silence, versus the MIP12-like silencing, you can see a clear decrease in flavonoid content in the silenced plants. We also try to overexpress the gene in a pink tomato background, so in a MIP12 mutant of tomato. And also here you see that when you have high expressors of the MIP12-like gene from pepper, you can see a clear increase in flavonoid content compared to the low expressors or the controls. And last but not least, very important for breeders, is can we use this gene actually to increase the flavonoid content of commercial materials. And we did that in a few backgrounds, and I'll show you here the results of introgression in sweet banana. And you can see when you introgress the P12 allele, you really get a nice increase in the flavonoid content in sweet banana compared to, let's say, the control without this allele. So also for breeding purposes, this gene is a very nice marker, and P12 is a very nice donor for this trait. Now, the ongoing research is actually focused on validating QTLs that we found in the GWAS in biparental segregating populations. We are mining these QTL regions for candidate genes and hope to identify and analyze candidate genes. We also want to improve our GWAS study because our study was really limited in the number of markers. I already mentioned we use 10,000 markers, but this is far too low for a GWAS study. And that means that we are certainly still missing maybe important QTLs because we don't detect the right alleles. We are trying to alleviate this problem by sequencing the entire core collection, and this is something that will be done in the near future. And finally, we'd like to also combine different data types. For example, we would like to combine our metabolites with taste, as I showed you, but also with, for example, shelf life data. Now, finally, I would like to come to the conclusions. I think that gene banks harbor a very wonderful resource of genetic variation that can be exploited by both researchers and breeders. The breeders' toolbox has really expanded in recent years with various exomics platforms, making it possible to have a much more detailed look and getting detailed insight in our traits. I hope I showed you that metabolomics is very useful to get insight in the metabolic variation in a crop, in the metabolic basis of traits, and in the genetic basis of traits. And finally, GWAS provides novel donors, QTLs and markers for traits of interest. But an important part, and that will take years to come, is the validation of the QTLs that we found. So last but not least, I would like to acknowledge the people involved in this research. From my own team in Wageningen, Yuri Tikhonov, he plays an important role in all the research we do. Jos Molthoven and Fien Meijer, two technicians in the project. Yi Wu, a PhD student who participated in this work. And then Richard Vinkers, Matthijs Brouwer, and Giorgio Tumino, who were involved in the statistical analysis of the GWAS data. We collaborated with people in France and in CREA to set up these GWAS trials, and with Ilan Paran from the Volcani Center, in which we collaborate closely in this research. And finally, all the partners that participated in G2P-SOL, who made it possible to have such a large-scale project going on successfully. And this is where I'd like to end my presentation, and I'm happy to answer some questions. Thank you.
Thank you, Dr. Arno, for the nice presentation. So, ladies and gentlemen, now we will have a question and answer session for about maybe 10 minutes. So you can deliver your question by chat box or raise hand. So please, you can start to give questions to Dr. Arno. Okay, now I have one from the chat box.
Yeah, very nice question indeed. Okay. For a human tasting panel, won't capsaicin ruin your taste bud to differentiate other tastes and aromas? Yeah, it's a very good question, and especially for Western people, it's of course very difficult to taste anything once you taste capsaicins. So if you talk about aromas, it's not so problematic because you don't taste it, but you just smell. But for really the combination of tasting and smelling, we cannot do that ourselves. And we did that in the past using actually Indonesian people to do the tasting, because they can much better differentiate tastes in the background of high pungency.
Okay, thank you, Pak Dani Satyawan from BRIN. Is there answer your question?
Yes, and it's quite amusing actually to find out some Indonesians actually have some use in this kind of studies. Thank you, it's very amusing.
Okay, thank you. Next, please, for the next questions. Or maybe I can also ask you, Dr. Arno.
Yes.
I'm interested when you said that metabolomics or metabolic is basis usually used for the complex traits. So what about the, like disease tolerant or resistant? Yeah, because we know in Indonesia that are the big problems, especially for this chili pepper. You know, Indonesia very love chili pepper. Chili fruit or chili pepper very well. So maybe you can explain about this. Can we use also this metabolics?
No, certainly this can be done, and we have very nice examples of it also. So also in the basis of resistance, metabolites play a role. And indeed, metabolomics can be used to get a better insight in the mechanisms that the plant uses for resistance against pathogens. And especially also when you want to, for example, get insight in the type of genes involved, a very detailed insight in the structures and the metabolites that are playing a role there, it can really help in identifying the underlying genes.
Okay.
Yes, it's definitely a very nice combination to combine resistance and metabolomics.
Okay, thank you, Dr. Arno. I see that Dr. Wahyuni is raising hand. Please, Dr. Wahyuni.
Yeah, thank you, Bu Hayati.
Yeah.
Hi, Arnold. Thank you for including my pictures in your slide.
Well, you made the basis for our paperwork, as you know. So
yeah, I always with your G2P Sol project. It's a very big project, including at least 400 capsicum annuum accessions, in addition to Pakatum, Chinensa, and other species.
yes.
And I saw the PCA of the volatile compounds. It's ended similar. I mean, the volatile compounds with this big hundreds of accession, they differ the accession based on the pungency level. Like we have in the last time that we used 32 accessions. Yeah. And then with this project, have you already identified if whether these volatiles came from or derived from the capsaicinoid pathway?
Yeah.
And yeah, the second
Yeah,
question or not? Or later?
Shall I first answer your question? Your first question?
Yes, please.
Yeah. Because you know, this is a very interesting question, because what we indeed see is that pungent accessions make a lot of volatiles, specific volatiles that you only find in the pungent accessions, and we don't know if this is, let's say, due to a really genetic, for example, the PUN1 locus, whether that is also involved in producing specific volatiles, because it's an ester. PUN1 is an ester-forming enzyme, so basically you could expect that it can also, for example, produce volatiles, or whether this is due to the fact that it's just an association, because these are two different sets of germplasm, the hot germplasm versus the non-pungent germplasm. Actually, we still don't have the real answer to that question, Juni. We had the question already in the time that you were working in Wageningen, but it's still an open question. And but we know what type of experiments to do to show it.
Okay.
So we really need a PUN1 to make a PUN1 mutant, an isogenic PUN1 mutant that in a pungent background, and that I think will enable us to answer that question.
Okay, so this one of the complex traits, right? These volatiles.
Yeah, sure.
Yeah.
Yes.
And
no, so we are very interested in answering that question, but didn't do the right experiments yet to answer it.
Yeah. That's one question that I want to ask you. If we have this complex traits, and can you suggest, or well, at least if you have something in mind, what kind of experiment that we should design to answer some kind of, for example, the markers underlying these complex traits?
Yeah, I think the problem with complex traits is that genetic studies are difficult because the complexity is too large, and you don't have the statistical power to detect all the players that contribute to your complex trait. So that's why we think it's important to really define subtraits, which we do based on, for example, metabolites, and then study each of these subtraits, because it will make genetics simpler. So that's one thing I would advise to do, because if you only look at the phenotype of the complex traits, probably you're not going to get there. So getting insight in your trait is very important, and so metabolomics is one way to do that.
Yeah,
is that answering your question?
Yeah, well, yeah, metabolomics.
And then of course, the next step is when you are able to identify, for example, markers for different of these subtraits, of course, you have to prioritize them based on the contribution that they make to the final outcome of your complex trait. And that is something you have to evaluate, for example, from your QTL analysis to see what is the percentage explained variation for each of these different subtraits. And then you can focus on the ones that are giving you the best possible outcome.
Yeah. Okay, Arnold, thank you.
Okay, thank you, Bu Wahyuni. So we still have now from the chat box from Bapak Dylan Lowry.
Let's see.
How would you plan to approach the unknown compound that detected in your LC and GCMS analysis?
So as I already mentioned, in GCMS, it's not such a big problem. We have only a few unknown compounds, but in LCMS, that's really a problem. Now, we don't aim to identify all these compounds. That sometimes it can take a PhD study to identify a few compounds. It can be very difficult. That's why what we do is we use these unknowns in our studies, but only when we can see that an unknown compound really contributes to a trait of interest, it is worth for us the effort to try to identify it. And the way to identify it, for example, is to do a much more elaborate LCMS study with MS fragmentation in order to really find out what the structure of your compound is, just as an example. But our approach is very on purpose. Don't bother too much about the identification in the beginning, but do that at the end when you know that these compounds are really important.
Okay. Mr. Dylan Lowry?
That was, I think, the answer to the question. Yeah.
Okay.
Then how would you know that the unknown is gonna contribute significantly to the traits?
Okay, so well, one way to know that is to do a correlation analysis to see if there's a correlation between the presence of your compound and your trait of interest, as I showed you for example, for taste. And when you have a certain taste attribute and you look at the correlations of all your metabolites with this particular attribute, that's one way to, for example, look at this correlation. But there are many different ways to do that. One other way that, for example, we like very much at the moment is when you are interested in taste and you run a GCMS analysis, one of the things you could do is to connect your GCMS device to an olfactory port and smell each of the compounds that comes off your GC column. And in that way, you can also detect specific flavors and detect specific compounds. And what we have seen is that sometimes compounds that are highly detectable and give you very small signals can give a very powerful taste experience, and also the other way around. So this is another way to find out, okay, I don't know the compound, but this one we need to focus on.
Okay, Dr. Arno, we still have one more question, and maybe this is the last question for this session from Dr. Penny Lestari, or Ibu Penny Lestari. So, on the level of pungency of chili is also influenced by the environment, such as dry and other stress.
This is an interesting question. It is known indeed that capsaicins are strongly influenced by the environment, and I know that breeders are very interested in ensuring that they have a stable amount of capsaicins in their crop that's less influenced by the environment. But we don't, I at least don't have the knowledge whether how big that influence is and whether this indeed changes with shelf life. It's a good question, but I don't have the answer.
Okay, so Bu Penny, maybe we can further discuss with Dr. Arno after this session. Okay, So thank you for the fruitful discussion. Oh, there is one more. Maybe Dr. Arno can answer with a brief question. These questions in briefly.
So which is the most reliable result between genetics and metabolomics? I don't know exactly what you mean with this question, because I think it depends. Reliable for what? So that's hard to answer. But both platforms have their pros and cons, and I think that especially the combination is strong. If we, for example, see that compounds that are correlating also end up at the same regions, also genetically show the same QTLs and so on, well, this gives us confidence, for example, that a certain compound may be involved in a certain trait.
Okay, thank you, Ibu
Yeah,
Rita.
so it's basically using the correlation on the one hand, genetics on the other hand. If everything points in the same direction, that gives you a certain level of reliability.
Yeah, but metabolomics also needs the genetics information, right?
Yeah, you know, when you want to do metabolomics and you would like to look at variation, or you need variation in order to see differences, and our approach is always based on detecting differences. And there, you know, having, for example, genetically diverse material is a must. Another way to create diversity, of course, is different treatments. We have focused much on using genetics, but you can also use treatments. But especially also in relation to the question of the identification of compounds, when you don't have variation, it's also impossible to, for example, prioritize compounds to focus on. So that, I think, discriminates also metabolomics from, let's say, the more classical biochemical studies that are really aimed at identifying every compound. But we are really focusing on the ones that do the job, that are important for our traits.
Okay. Thank you for the questions. So, and also this fruitful discussion for Dr. Arno. So maybe in the future, if we can see from your presentation, this metabolomics and GWAS or genome-wide association study will be more improved, and many people maybe use it for in breeding session, right? Okay. So again, we appreciate Dr. Arno Bovi for the excellent presentation. Maybe by giving him a big applause. Thank you for your attention also, everyone, and I will close this session. For the next program, I'll return to the committee or the Master of Ceremony. Thank you again, Dr. Arno.
Thank you too. It was a pleasure.
Thank you.
Thank you, Dr. Arnold, for the interesting materials. And also thank you to Dr. Hayati for leading the sessions this afternoon. Very fresh, ladies and gentlemen. So for the next agenda, we are going to announce best oral presentations. Who is the best oral presentations during 2 days of ISIBio? Any guess, ladies and gentlemen? And the best oral presentations goes to, drum rolls, any guess? Yes, congratulations to Dr. Sugiono Saputra from Research Center for Applied Zoology with the title of Sago's Chart Hydrosate as a Carbon Source for Expolysaccharide Productions by Lactobacillus plantarum. Congratulations for Dr. Sugiono as the best presenter at the NIN International Symposium of Innovative Bioproductions for Biotechnology and Bioengineering, ISIBio. Please give a round of applause, everyone. Ladies and gentlemen, and now I would like to invite Mr. Ahmad Fatoni, PhD, Head of Research Center for Applied Microbiology, National Research and Innovation Agency, to convey closing remarks. Bapak, screen is yours. Still unmute.
Yeah, sorry. Yeah. Sorry.
Yeah, thank you, Ms. Aulia. So first of all, I would like to thanks here still we have our speaker, Dr. Ahmad Bobi. It was very interesting talk, and then many questions. I hope that it can be follow up by the collaboration with him in the near future. Okay, everyone. Assalamualaikum warahmatullahi wabarakatuh. Good evening, grace and peace, everyone. I'm Ahmad Fatoni, representative of Dr. Iman Hidayat, Chair of the Research Organization for Life Sciences and Environment BRIN. It has been a pleasure to be with you since yesterday in the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2022 with the theme of strengthening bioeconomy through applied biotechnology, bioengineering, and biodiversity. Thank you everyone again for attending this symposium. On behalf of the Research Organization for Life Sciences and Environment BRIN, I would like to express gratitude to all keynote speakers, invited speakers, presenters who have shared their idea, knowledge, and finding with us during the symposium. Since yesterday, I observed the excellent presentation from the great work of everyone and active discussion among our speakers and participants. So this symposium covering 6 topics, including bioresources and biodiversity, nutraceutical and natural products, biotechnology and bioinformatics, medical sciences, agricultural and forestry, and the last, environmental sustainability. So therefore, I can conclude that the purpose of our symposium this year has been accomplished. But however, I hope that this symposium is not just a tool for us to share our finding or idea and stop with the publication, but bear in mind that we still have a big responsibility to implement our findings to our any potential collaborators or stakeholders such as society, industries, and other users. Hence, our research hopefully can make a significant impact to the economy, society, and the nation or Indonesia. And one of the key success to achieve enough impact is collaboration. And I would like to take this word of collaboration as a key home message to all of us. Implementing our results and making an impact are not easy tasks, but I believe we are going to make it by working together with others. As our chairman of Research Organization for Life Sciences and Environment BRIN, Dr. Iman Hidayat mentioned yesterday in the opening session, that there are 3 factors that we should have done. Actually, first, working together with colleagues from other disciplines. The second, working closely with industry, and the last, mastering key technologies in order for us enable to utilize our biodiversity sustainably. So I do really hope that after this symposium, everyone, we would extend our collaboration with other colleagues that we just met during this 2-day symposium, or even with our keynote speakers from Indonesia or overseas. And here we still have Dr. Bovi. I hope that in the near future, or Dr. Wahyuni already have collaboration with Wageningen and others. Hopefully we can strengthen the collaboration with the Wageningen University. And I believe also that researchers and participants here already have their own collaboration, but I think there should always be a room to enhance our collaboration. Okay, before I close my talk, congratulations to Pak Sugiono, who have won the best presenter, and hope it will give motivation to us to get better and better in the future. Lastly, I would like to thank the entire committee of ISIBio 20. Oh, sorry, stop my video. Yeah. Can you see my video? Yeah, lastly, I would like to thank the entire committee of ISIBio 2022 that have done a fantastic job. They have spent their time, thoughts, and energy to prepare all of this. Thank you, Dr. Nani, Dr. Wahyuni, and all team members for making this symposium a successful one. Also, thank you to our collaborators, Indonesian Biotechnology Consortium, Consortium Biotechnology Indonesia, of Indonesia, and other who have supported this event. Again, to all participants, thank you very much for your attending in ISIBio 2022. And finally, I would like to close my remarks and officially end the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2022. Wishing you all a success and hope to see you in the next ISIBio next year, inshallah. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Stay safe, everyone. Thank you.
Thank you
I
for
give
the
back to
remarks. And also officially closing ISIBio 2022, Dr. Ahmad Fatoni. Ladies and gentlemen, that was the last agenda of these 2 days, ISIBio 2022, as well as mark the end of ISIBio itself. On behalf of official committee, I would like to sincere highest appreciations and also thank you so much for your attendance and active participations in all sessions during the conference held. See you on the next ISIBio next year. Stay safe and healthy.
Okay, thank you, everyone. Yeah,
Thank you.
do all the best. And see you next year. Yep. Thank you.